Hola, eco, y soy bastante divertido. Te he cortado, me parece. Ah, no. Te hemos hablado un poco estresado. Ahora. Hola. Buen día a todos y a todas. Então, segundo dia da escola da Spring School, de aqui de Fortaleza, aqui com essa mistura também de pessoas que vêm da Europa, pessoas que vêm do resto do Brasil, pessoas que vêm também de Chile, para fazer. Hoje tendremos, muito importante, então, a terceira charla, a terceira palestra que temos, não pode ser feita. Vale, volto un'altra volta a empezar. Voy a falar un momento antes, mientras estamos aquí conectando tranquilamente. Lembrem, agora passaremos a lista para que vocês pongan seu nome, con nome, a seva presença. Lembrem que os que não estão filmando presença não têm curso. Os presenciais, obviamente. Os de online están na outra parte, é un método diferente de seguimento. Okay? Agora passaremos a lista para que vocês possam seguir. Io penso che una vez che vocês hayan feito el check-in allá como persona, que pues vocês tienen que firmar cada día como... No, no, mañana está bien. Mañana está bien. Esto hacemos nosotros aquí, mañana tranquilamente, ¿vale? Então, eso por una parte. A otra, vamos a esperar, porque a otra es también para las personas online, ¿vale? ¿Ya podemos? Yo estaba allá en la mesa, pero estaba aquí dentro. Penso que aí vocês que se inscreveram. Ok, vamos a ver. Entender, esos son dos listas diferentes. Una lista es para os estudiantes de posgraduación, que son esos que habían algunos estudiantes que no estaban en la lista, pero que se habían matriculado y que tienen que añadir un nombre aquí y poner firme y tal. Y eso es para los alumnos, otras personas de graduación o que han venido de otros departamentos, donde tienen que poner nombre con nombre y la asignatura. ¿Vale? O sea, uno es de posgraduación y otro es de otras personas que se inscribieron, o sea, directamente pues, y que no están vinculadas a posgraduación.
Puedo comenzar a falar e aí eu
Otra vez, vamos a voltar a comenzar. Perdão, sobretot para os que estão na outra parte da tela, as pessoas que estão na outra parte do resto do mundo, desde Sérvia, Turquia. Eu sei que há pessoas que estão na França, Bélgica, Alemanha, ¿vale? Perdão, porque temos surgido um pequeno problema técnico com a câmera, mas agora pues já está resolto. Então, bem-vindos uma outra vez aqui a Spring School, a la Fortaleza Spring School, que promociona a LABOMAR y la Universidad Federal de Ceará, que está apoyada directamente, obviamente, por el proyecto COST, la Cost Action europea. Es una mezcla de las dues, una sinergia entre las dues possibilitats, las dues, digamos que interacción y parte de internacionalización y networking. Es una cosa muy interesante, con un apoyo de Air Center, que está directamente desde Portugal, nos está ayudando en la parte de streaming. Entonces, cosas importantes. Hoy, a tercera palestra, la profesora Federica Costantini no va a poder ser. Ella tuvo que voltar al domingo por problemas personales en Italia. ¿De acuerdo? No tenemos esa palestra. Más tendremos un vídeo de unos 15 minutos del profesor Lorenzo Bramanti sobre restauración de coral vermelho, muy interesante, y un pequeño vídeo también de 1 minuto, donde vocês podrán ver esas transiciones das Marine Animal Forest ao longo de un gradiente de profundidad. ¿Vale? Entonces, sin más demora, vamos a comenzar con el primer palestrante. And the winner is el Dr. Américo Montiel, professor, is a biologist from the Universidad de Magallanes with 28 years of professional experience researching marine ecology in Antarctic and subantarctic areas, with special emphasis on biodiversity and taxonomy of polychaetes, with a PhD at the University of Bremen, Germany. He has participated in numerous international scientific expeditions, sailing abroad the research vessel Polarstern, I know because we were there, in Antarctic waters of the Weddell Sea, Bransfield Straits, and Scotia Arctic Sea. He also organized and participated in several national expeditions to Patagonian fjords and channel waters. Also, aboard of research vessel Son, he has participated in expeditions to study the benthic upwelling system in the northern Chile. Please.
Well, thank you, Dr. Rossi, and thank you for all the people that coming to today to see my presentation. Could you please say from the beginning? Yes. Now I have the power. Well, today I want to speak about some many researchers that we make in the last couple of years. I entitled all this investigation under the title Animal Forests at the End of the World and Beyond. So I need to thank to the organization, to everybody to invite me to present this information. So during this presentation, I will show 5 different history, some from the end of the world and from the Antarctica, and with the old-fashioned nomenclature, the end of the world and the Terra Australis Incognita. So, one of the important things in every presentation is to give a context of where we develop all this research. In this planisphere imagine, we can see where are the old fjords and channel ecosystem. Obviously, in the northern hemisphere are most ecosystem of fjord and channel because are more land, and all these fjord and channel were developed geologically during the last maximum glaciation. So this is the line where there was the ice in the past 10,000 years ago. No? And in the northern hemisphere, where was more land, were more erosion, so it's more ecosystem in the northern hemisphere than in the southern hemisphere. So we are lucky in Chile that we have the largest fjord and channel ecosystem in the southern hemisphere, together with New Zealand and obviously from the Antarctic Peninsula. So some important numbers of the Patagonian fjord and channel is that they are more or less from the beginning, more or less 42 degrees until the 56 degrees south, and this is more or less for 14 degrees latitude. This cover around about 240 kilometers of area. And one important thing, if we make the straight line from the beginning, for the 42 to the more or less Puerto Montt city until the Cape Horn, in the end of the South America, we have around about 1,500 kilometers in a straight line. But with all these different fjord and channel, the coastal line increased 56 times. So this means that our coastal line in this ecosystem is 84,000 kilometers. This for that you have a relation. Brazil have 7,500 kilometer coastal line. So it's many times more than coastal line that has Brazil. In Patagonia, we always say if we take all this in a line, we can give 3 times around all the globe, or the Earth's globe, with our coastal line. So in this area is many different kind of habitat, different kind of situation, local situation, with different estuarine area, marine area, together with many of the ice sheet or ice field that is here in the Patagonian. There are at least 3 ice, the North Patagonian ice field, South Patagonian ice field, and the Cordillera Darwin ice field. And many others, small ones, that Gran Caponegado, that is as a joke name. This is not the biggest, but it's called the big one. In this large ecosystem, we have many latitudinal gradients that we show just 2 examples here. just the latitudinal gradient in terms of temperature and the salinity. In all the imagine of this is red and blue, and so we need always look the scale. So the more hottest is around about 15 degrees Celsius, and the coldest is 0 degrees. So in all this field we can find all this gradient of temperature in the is enormous from the 15 to, and obviously it's cold in comparison with what we have here in Brazil. And here we have this from very warm 15 near to Chiloé Island, and in the Cape Horn is close to 0. The same with the latitudinal gradient in the salinity is more come from 33, the unit from the salinity, PCU, and 54.5 is the highest salinity. So if you look in the latitudinal gradient, we have low salinity in the closer to the ice sheet, and less salinity, increases the salinity here in the south part, and both are changing in the depth. So this is more or less 300 meter depth, so from 0 to 300 water meter depth. So this is more or less the situation. Not only latitudinal, or also depth gradient, and also longitudinal gradient of temperature and salinity. And we can say from the colleague of the oceanography, many nutrient, phosphate, silicate, and so on, will be all show some gradient in this area. But more or less, this is how it looks like our study area. Now this is a picture from the airplane. If you sometime go to Patagonia, if you have luck, have a blue sky in this moment, you can see this picture with fjords, ice, and channels, and bays, and all this is a estuarine area with seawater, or breaking water and seawater in the bottom. So this also in many places is these glaciers, and they discharge a lot of sediment in the water column. So this is the reason why it's so this green water, very turbid. So this is the closest part of the many fjords and in our Magellan region. So this is a cartoon, try to show a little bit how looks like this, or try to put together in the context that we have a strong relation, land and sea relation, and also with the atmosphere. In the atmosphere, always raining, or for example, in one point, evangelist like raining 360 days per year. So it's almost all the day, every day raining in this point, and in the more highest temperature, snow, and this maintain the glacier or the ice sheet that I say previously. And this make a strong relation with discharge, sediment, and in because of the global warming, always some of them retreat, so make open new area for the colonization. Another make more thin glacier. So it's very complicated to make a general picture in Patagonian. Also, we can say two kind of fjord. One fjord is direct Calvin Klein fjord with a glacier in the top, like this. Like this. And also there are another kind of glaciofluvial fjord where the glacier was retreat and make a river, and the river discharge in the open sea. So they have two kind of influence of glacier. So and this makes this a strong estuarine marine ecosystem. Another picture make a little bit in a comparison between a normal situation, say normal in the Patagonia is very complicated. Some normal situation in the and within a situation with salmon culture, for example, and how function the system. So here is a strong capture with, of CO2, make a phytoplankton, load this layer of brackish water that exists in the Patagonian, that in some area could be very thin, something like 1 meter, and in another places can be 2 or 3 meters of water depth. And from this brackish water, when we dive, we saw very clear because it's very turbid, and after that come very clear seawater. And in this area, we'll make the normal thing, phytoplankton, krill, and make the situation with the deposit and flux of carbon until the bottom. And with the salmon culture, make something different. So the capture of CO2 decrease and make all the different, change a little bit the situation that is in the normal situation without a anthropogenic impact. Yeah. So how we study this very heterogeneous ecosystem? So we need to have many approaches, not just one. We make a diving, take picture, but also in the bottom or in the bottom of the glacier or in the bottom of the fjord. Sorry. Exists a lot of soft sediment, so we need also to make drag or grabs or trolls to capture all the biodiversity and look how work in the benthos ecosystem, and also with some technology with the ROV and looks in this. Then with the time become more common the picture and also from the ROV because the technology come a little bit cheaper, so we can make more investigation in this way of with this approach. After that, we need to look in the laboratory or make like multifunction looks, video, picture, and so on. Yeah. So all in one picture. So all this cartoon is using this imagination or it's very synthetic. All these things in this picture we can see what I showed in the previously river. Behind here is a glacier. Here is the rocky bottom or rocky reef. Some large animal forest. With the bivalves, the Mytilus chilensis is the dominant, or Aulacomia ater are very large blue mussels, and also the kelp, yeah? The macrocystic peripheral. This other common organism in the fjords. So, and to arrive to the main things of this presentation, first I think it's very important to have a theory also about where come from this all this idea of animal forest, or how we say the functional ecology. Yeah, this is not so new. Here is the Professor Charles Elton in his master book, Animal Ecology. He described for first time that idea of that is important to see or looks like the function of the animals. This was the first approach to then develop this idea in what we know now like a niche and dominate for the theory of Hutchinson, that hypervolume of the niche, but the original idea was from the Professor Elton, and say, looks the function more than the numbers. Yeah? This is important. So there is the definition of the functional ecology. Yeah? And in this way, or in this frame, Professor Rossi developed this idea of animal forest that looks the function, and the function is more important than just to say one number and so on. This I will show with the following. So more than the 1 and 0, the traditional ecology make values of this one species is 1, this is a 0, this is 1, 0, 1, 0, and make a nice statistical or make abundance and so on, but don't take account all the forms, yeah? The shape, the difference. In this picture you have many shape, many form. In the trophic gill, for example, also maybe it's just one trophic gill. This is suspension feeder. Or it are detritivore. So in this picture from this is a typical bottom of the Patagonian because it's dominated by this coralline incrustant algae, and this and all make a little bit pink everywhere is dominant in all the picture that you have. You can see in the research that will be shown in the one moment. You see everything is pink, yeah, and this is also we try in the future try to calculate how many is the carbon that collect all these. We don't have corals or not have so large coral that here in the tropic, but we have the coralline algae and also all the other animal forests. So then began the first history, how approach to the animal forest. This was 10 years ago. Was the first idea when Professor Rossi invited to me and also to my, in this moment, student, Cesar Cárdenas, to publish it a little bit our result that but with the view of animal forest. So, and we develop a chapter inside the book. So the coexistence in cold water, so animal forest, we seaweed dominate habitat in southern high latitude. So this is our chapter there, and this was the first approach where we introduced the idea also that is important that the animal forest not live alone, also live together and coexist with many other species. Yeah? And not always is in this way is the dominant animal forest, but are the second important in terms of cover or in terms of abundance in the bottom, in the rocky bottom especially. Yeah? And well, that you know, we dive. You see pink, pink, pink everywhere, but between the pinks are the animals and rocky. This picture in there, I am there, yeah, with the reggaeton belt, very was properly used. Always I have this problem. But anyway, we take the information and we take the pictures, and you see here is many animals, animal forest, the coralline algae, and another errantia animals. No? And we make to make this approach to look the coexistence of both animal forests and the seaweed. We look with the following this. There is the macroalgae example. We have Lesonia, Adenocystis, and another red algae. Let's change the name. For this reason, I don't remember exactly. Yeah? But we looked, our approach was to look in the shape, how is the growing the animal forest, as well as how is growing the macroalgae. And we found the similarity in both. Some of them growing in a tree form, so with looks like a tree. Yeah? Also, another growth form was the arbustive. So the arbustive is a bush. Yeah? It's more without the stall, the definition without a stall, and make a bush growing form. Mound, that is something like a sponge, or the other picture is a round thing. Yeah? That is also a kind of grown form. Foliaceous, yeah? With a leaf and laminary, that is a sheet. Yeah? This is growing incrustant in the animal form. All these 5 form of growing was similar in animal forests as well as in the macroalgae. Yeah? And then began to compare both in our pictures. So, and also some of the main factors that drive the distribution, the animal forest and the macroalgae, are the inclination. So they have, we test 3 different inclination of the bottom: inclined, vertical, and overgrowing. This should say here in this table. So this is inclined, vertical, and overgrowing is something like in the shadow of the area. So and make some graphic about that. So the animal forest and with the different kind of growing forms. So bush, mound, and trees, the animal forest, and here is the bush, mound, and tree, but the macroalgae forest. So, and we saw immediately some difference. Here the macroalgae growing mainly in the inclined surface of the bottom, those the rocky bottom, and the animal forest, especially bush and mound, growing better in the overclined. This make a little bit thin because in this area the light of the sea, we're talking about between its diving depth, so something like between 5 meter depths and 30 meter depth. So the light, the sunlight come to the surface. So the macroalgae prefer this kind of inclination better than the overgrowing that is in the shadow. So, and this take the opportunity for the other organisms like animal forest, and growing here, and mainly filter feeders are in these things. The trees show something special, growing also in the inclined and vertical, and also in the overgrowing. That is more or less detritive animals, so can live better in all the situation of the inclined of the substrate. So, second history of the animal forest is we make an experiment to test this. We saw in the nature, and we try to see what happens if make a coral reef, artificial reef. So we developed this idea. Make this was still this is before pandemic. The idea was the project was in 1990, but we put in action or put the coral in January of 2020. The idea was to follow every month how they colonize this artificial reef, but with the pandemic in the 2020 was not possible. So I will show just the beginning and 1 year later. when we can go again together in a trip and go to them in the sea. So, but we developed this kind of coral artificial reef. There is a mound with owls, and there is the planning, how the something like 1 meter, 3 meter diameter, and run about 1 meter 20 of high. So there is how it looks like. There is in the harbor, and put in a ship and go to the sea and put in a sand beach or sandy bottom to evit that the colonization could be not from the bottom to the artificial reef, so come from the larvae. So this was the idea to put in this substrate. And how we saw was success. The first thing was here is I don't bring a video because I make sometimes problem with the video, but I have the video of the 1 year later. So was colonized successfully for many organisms, and there come again the coexistence between macroalgae and animal forest. In the surface, all the surface out of the artificial reef was full of algae, including macrocystis and red algae. More or less was something like 20 species of algae colonized in 1 year. In the surface of these three how to say, prototype of artificial reef. Also was another crab because it changed the current, so it was protected, and was many of these kind of shrimp. So it's Munida subrugosa, this species. And obviously, again, our corallinian algae, but inside the artificial reef was full of animal forest again. So it was sponges, Irudinian, and many animals was in the shadow or inside the coral artificial reef. Artificial, no? And so there, we saw that the inclination was also important, or what we saw in the nature was tested in an experiment, and we see that the vertical surface was very nice for the algae, and the more shadow area inside the coral or the artificial reef was better for the animals. So there I have some. In one moment will be published the data with the Sergio. I hope this year. So, and there is more picture about that. Not only the biggest guy with the animal forest, also some small one. And one of this was surprise for me was that this was just 1 year. So in 1 year was full of life and with a lot of capture of carbon, because they are also animals, these polychaetes serpulid, they also have tubes from calcium carbonate, and was full of them in 1 year. And this was no water from the bottom, so was just arrived by larvae and colonize all the three artificial reefs that we put in close to Punta Arenas. So this is then we go to the first history from animal forest, and this is from work from colleagues from the Winai Center, and that was also published in the book from Animal Forest. But they make another approach to categorize the animal forest. They use classification made by Jackson in the 1977, and they use how is the chemical structure of the body of the animal forest. So some of them are with calcium carbonate, another are psyllium or mucus with sand or chitin system, all a different kind of chemistry, how built the structure of the animal forest. So there is a sample, taxon, and also include this solitary or colonial or different kind of categorization, and by the different taxa. Here is Anthozoa, Alcionaria, Zoantaria, Scleroctinia, obviously are solitary, for example, another are colonial, and so on, and make this with this classification. So, and they show that in the animal forest in the Fjordan Channel, there are 4 groups. One is conformed by bioengineer or calcifying bioengineer. That is many of them, and this is very important when we saw, or keep in mind when we saw in the Antarctic. This kind of animal forests in the Fjordan Channel are very abundant. So in Fjordan channels, there are a huge, we can say the main benthic marine resource are mussels. Yeah, we have Choro Zapato, for example, that is some large one, so like 60 centimeter long, the biggest thing. Yeah? And also Aulacomia ater, yeah, that's is less bigger, but let's say something like 40 centimeter length, and Mytilocchilensis, that is blue mussel, smaller. Yeah? But when we saw this in terms of how many carbonate accumulate, is a lot. They take tons, something around 3,000 tons per month from all the Patagonian Fjordan challenge, from different areas. So some of them are eating in the local market, and others are sending to different parts of Japan, Chinese, because you know this is the main market. And this is many common, the mussels in the Magellan area, but also there another one, Anthozoa, Scleroctinian, that build external skeleton by carbonate. The another one is there is also the make calcifying bioengineer presenting a massive endoskeleton. There is, for example, Erina. This is one of the cold water, the most famous cold water coral in the Patagonian area. This and also it extends on some of them, but not so abundant in the Antarctic. So, and also bryozoan, make a very bush of them, and this is make only carbonate. So we in the deep, we in the fjord store a lot of carbonate by this animal forest. So to look a little bit from the importance of this animal. So another, this make calcifying bioengineer, but with scattering calcified structure like the different soft coral. So there is this was in the moment a new species or coral latigo from the Coccinia, I think is the genus, and Gorgonian. So all the people that know Gorgonian have is that you cannot take it and break it, so it's very hard. Some of them you can took and take. You cannot take out from the stone, so fixed in the stone. But this is because the structure of the animal is very fixed in the depth. Another no calcifying animals, so like polychaete, for example, this is papiro polychaete, this is a Ketopterus variopedatus, yeah? And make also in a very, in a, no, it's not, something like a colony, yeah? But every tube is an individual, yeah? And between all these tubes, growing algae, growing another animals, also inside the tubes live some crabs inside, or other polychaete also live inside the tube. So make what one marine animal forest need to make. Yeah? And also this is oops. Another thing is what is urocordata, that's growing in the Magellan region that we can also eat. They have a very special taste, yeah, but you can make soup and so on, yeah? And be a tunicate, so make all this surface, and inside between them growing a lot of animals. So, and the forestry is going a little bit forward to the crossing the polar front. Yeah? What I can say this? Oh, sorry. Okay. Yeah. And this, before that began with the history of animal forests, was a publication of Nuria Texido, a colleague that also Dr. Rossi was in our time that we was together on Polarton. So they make also this kind of grow form, bush, sheet, tree, and mound, yeah? And make the comparison how the growing form in the Antarctic area. So, but in this moment, was the central question. The question in this moment, between 2000 and 2005, in the moment was the research was focused in the how recover the sediment in the Antarctic area. Yeah? Because was a lot of disturbed by iceberg. So, and they put attention in this more than the how growing form, but uses this kind of approach in the 2000 and looks. Here are four pictures. One is in the this is is ordered in a successional time. This is direct, more or less, after iceberg disturbance. This is the first colonization, second colonization, and the climax community, where it's full of animal forest, spongy, bryozoan, hydrozoan, and so on. So this was one of the first approaches. Obviously, it's not called animal forest, but use the functional ecology. And there is some analysis, so there is different kind of in different situations. So also different, the growing form is very important for the animal forest. So it's not just all the animal forest, it's just one. So depend of the growing, and also in the Antarctic we don't have so much difference because all the coast is covered by ice, obviously. And there is more or less in the ice continental shelf, so it's more or less flat, yeah. And they make a difference by the growing form. So another example, and what my final history is about, like the animal forest can be also considered a pioneer of colonization, yeah, after a recent deforestation area. And this was how we do in February from this year. So the analyses are in develop. So I hope publishing this in this inside this year. But I will show some highlight about how it looks like in the glaciated area in the Bellinghausen Sea. And this is the chip, this is the Falcor. Where we go, there's a icebreaker class C, so it's not like polar sand that is class A, that they can break, I don't know, 1 meter depth ice, so this can be go carefully inside the Antarctic water. Yeah, but we have luck because in the beginning, the plan was to investigate the canyons here. But in the 15th of January, or 25 of January, was informed to us that a large ice sheet was break and go away, yeah, or drift away in the Bellinghausen Sea. So we change the plan and go direct to there. So there is Sebastian. This is the main gear of the Falcor. Is a ROV of something like 2.50 m high, 3 meter long. 5 tons of and have many arms and CTD, current bathymetry, and so on. So they have the ultimative technology have this ROV. So we go there. Here is, sorry, there was the 11th of January. We was just go through the Drake Passage and inform us, and we go direct to this area. So here is this is the large ice sheet that is break and drift away. So we go to take a look how it looks like all the bottom in the after the break this ice sheet. Here is an example how the profile of the bottom, and this is the more or less the transect that we plan, and we take this. And how what have this to do with animal forest? That is animal forest. So we saw the stones, many of them, and full colonized by spongy, silicate spongy, gorgonians, and this is in the bottom. Always we began the transects in the bottom of the 1,000 meter, 2,000 meter, and go up. And then this is the soft bottom. Well, have many hours of many soft bottom that we don't have so much, but in between appears this. But then began to the slope of the canyons inside there, and we found more animal forest, more spongy, more than them. Oh, the other way. And in the vertical, again, the inclination make an important factor. So there is many sponges, more gorgonians in this area because the current water goes through and make the resuspension of the sediment and the food of the animal. This make food for all these suspension feeders of animal forest here in this area. Another one, and dominated by a spongy. You see, this is and this. This ice sheet was stable for many years, so below or underneath of this ice, the current come just vertical, so this make all the food for all this community of animal forest. So more examples. They are not only spongy, also another soft corals. And to final discussion and a synthesis, we published a little about this drawing that in our chapter with Cesar Cárdenas, and we proposed as a hypothesis that most of the fjord and channel are dominated by gregarious growth form for animal forests like bivalves, and in the Antarctic ecosystem are growing more by clonal and animal forests like spongy, like I show in the picture. So I can also yesterday, Dr. Rossi also showed the colonial of large field of spongy, silicate spongy, and so on. And in our the research that was in January of this year showed that this again is dominated by sponges. So this is not just a puntual or local situation. So it's something common in the Antarctic. And maybe the possibility of this different trend is because the calcium carbonate solution increased with the water. So this is the reason why in the north part of the polar front, fjord and channels, we have these large carbonate animals, so the mussels and the coral and so on. And despite in the Antarctica, or in the contrary, in the Antarctica, where the water is colder, the carbonate calcium is difficult to catch by the animal. So all these big mussel cannot grow in this so cold water. And this is the reason why another animal forest dominate in Antarctic. And also important thing is that the slope or the inclination of the situation is a dry distribution in this area. And like a future perspective, I will be happy to make some more close analysis, like I showed in the presentation of yesterday, that with different kind of measure, or is one of the wish that we can say that is mapping so the animal forest along the fjord, because we have some spot, but when we mapping all together, maybe find another kind or more general ecological pattern in fjord and channel about the animal forest. So with this, I want to thank to organize to Dr. Rossi and the director of the LABOMAR that was concentrated on another thing, but I'm not catching the picture. And thank you very much for your attention and question.
Thank you, Americo. Very interesting, and also, you know, in the other side of the world, you know, I think that the fjord area is one of the most complicated to study. When you compare the thousands of coastline, and yesterday we were talking about mapping, for example, imaging, for example, what means to have this approach in these areas, making a, you know, scrupulous and very, you know, fine-tuning mapping of these areas could be decades or whatever, I don't know, with the actual. Do you have questions? Some question? Pode fazer em português, se pode.
Primeiro, parabéns pela apresentação, foi muito boa, parabéns. E eu queria perguntar sobre espécies invasoras, como é que vocês lidam com isso? Porque aqui no Brasil é um problema já há muitas décadas, e principalmente nos organismos bentônicos, né? Poliqueta, corais, enfim. Eu queria saber como é isso lá para...
Si hay
well, I not saw personally, but many of my colleagues that dive in the north part of the Patagonian area say that is a white actinia that began to colonize the sub-bottom. It's very area strategy, so colonize very fast space and so on. This called something blanquear, something blanking. No, this is one of example that in the Patagonian and in the Antarctic. Well, that is always was I remember the Sedentary idea that that was the recolonization of the king crab. The king crab that was in Antarctic Reptantia decapod. And now we found also we found, I'm not sure this picture, sorry. We found king crab in the Bellinghausen Sea. So this began to be off the king crab come back to the Antarctic. This will be one of the colonization. Well, it depends on how you speak. You can say that it's an invasion. Also, another king crab was discovered, but it's a small one. It's a Licarcinus planatus that was found in the intertidal in King George Island. But for me, always was the question: well, is an invasion or really was there all the time, and we don't see before. So this was, and the king crab we saw in the Antarctic. I showed to Gustavo Lowrich. It maybe is a new species. So this was, well, some of the discovery that we make with the falco. So this, I think, this is are the another thing, but non-animals, is some algae that drifts. The Lesonia is a special. It's like the macrocystis. And also drift with a lot of animals inside in the foliar area, so they can also arrive to Antarctica by the current, the circum-Antarctic current, and can also arrive in the Antarctic area. So there is some approaches to the invasion, and also with the change of temperature and so on. Can be more species from the north part of the polar front can tolerate the change or inside the Antarctic. And obviously the Antarctic species that need colder, so need to be back to the continent. So there is some ideas about that.
You also have to think. Você tem que pensar uma outra coisa que é muito importante, que o professor está falando esta manhã. Não há pessoas allá e não há recursos. Então, fazer seguimento das espécies invasivas é muito complicado, porque há muito pouca gente allá trabalhando e ficando no mar, mirando, observando, tentando. É muito complicado. Eu me estava explicando das pouquíssimas pessoas que trabalham nos 27.000 quilômetros de linha de costa que tem os fiordos. Há zonas que ninguém ha visto nada nunca. Então, claro, e pode ser que estén chegando espécies invasoras de diferentes pontos, mas não estão registradas, porque você não sabe. É uma coisa complicada. More questions? Do you have more questions? I have a couple of questions for you. One, the question, very interesting. I love that you show me that graphs about the biodiversity, three-dimensionality, the place in which these organisms, both algae and animals, sessile animals, are just. Could you make some first approach about the associated fauna, zooplankton, for example, to these areas? Let me explain. Recently, we published a paper in which we do have an area, an integrated multi-trophic aquaculture, mariculture facility, in which we have no animal forest and animal forest. In the animal forest, the zooplankton was much, much more abundant than in the other place, in a seasonal cycle, and also during an intensive cycle. Could you make some first approach to this associated fauna, like zooplankton, copepods, or whatever?
With this, good question. I think that the most of the animal forests in the fjords are more related with ardentity. So it's more related with something like the relation with the sedimentation of the glacier. So this is a longitudinal gradient. So there.
Like that is just we can whip out a lot of this carbon that is fixed in the sessile organism. The question is, do you think, but this is I don't know, if there has been an increase in the ice scoring because there are more icebergs in Antarctica because of climate change? There are more icebergs, and then the risk to have more ice scoring is more evident.
Yeah. Bellinghausen Sea, and this then increase the damage or the scoring of the bottom. Yeah? Then this is in a theoretical point of view, yeah? Because we don't know how deep are the iceberg. So not all the iceberg produce scoring. So but it will be depend if the iceberg will be depth and where is the topography of where is become this collapse. It's not so easy to answer. But from the theoretical point of view, yes, will be increase this.
Okay, thank you. More questions? Not for the yeah.
Bom dia, né? Parabéns pela apresentação, um tema realmente muito massa. Primeiro contato que eu tive com essa área. Então, a minha pergunta é mais a respeito sobre os recursos minerais mesmos e como, na perspectiva de mudanças globais, né? Mudanças ambientais, isso poderia comprometer a biodiversidade marinha na região. Ou se você já tem uma perspectiva de estudo de, tipo, remediar esses impactos.
Yeah. There in the well in the marine biology is not a black and white answer. Yeah. I have some approaches not with the animal forest, but for example with the giant kelp macrocystis in the Patagonian. Despite all the changes of temperature, the heat wave that describe the oceanographer around the world, in the Patagonian are a refuge. No change too much. There was a publication we make with a colleague that the kelp forest in the Patagonian area is in the same position that was described by Darwin 500 years ago. Darwin make a very description in the Voyager, say here is kelp, here is kelp. We saw in the same place, and there is still the kelp. So this is one of the bioengineer, and in the term of marine animal forests, so I think it's more or less the same. We are happy that inside the system in the Patagonian fjord channels still a refuge from the world warming. Not change too much in this point. But in the Antarctic, it's a complete different situation for the scoring, change the temperature, and so on. And again, one of the things that we talk in and you also mentioned, this maybe is a problem of observation, because in all our ecosystem are run about 100 people, 100 scientists that study the sea, from oceanographer until people that study polychaete like me. So it's few people that study the marine system in the area. So in the data show that not so much change, but maybe if we have 1,000 people working, maybe detect the changes. So this is where I can say for my it's no answer, yes or no, but it's a maybe.
Thank you very much, Dr. Montiel. Okay, we pass to the next one. Which is Professor Martina Copari, which is a marine biologist. Her research primarily focuses on the ecology of benthic suspension feeders. She obtained a PhD in Environmental Science and Technology in Autonomous University of Barcelona in Spain, studying the importance of benthic suspension feeders in the biogeochemical cycles and as carbon sinks. During her PhD, she learned and applied remote operated vehicle imaging and video analysis to characterize the spatial bathymetrical distribution and population structure of different benthic suspension feeders, mainly sponges, ascidians, and gorgonians. She performed several in situ feeding experiments to estimate the role of suspension feeders in the benthic pelagic coupling processes. Her research is focused on the characterization of the ecology of cold water corals in the framework of the BioMount project funded by the MIUR-CIR in Italy. She leads the experiments involving isotropic diet, reproductive traits, bacterial associations, and growth. She's from the Università Politecnica delle Marche in Italy. Thank you.
Good morning, everyone. Thanks to be here to listen to my talk, and thanks, Sergi, to invite me and give me this possibility. So I actually prepared 2 slides to introduce myself, but Sergi already said many things, so I'm going to be really fast. I am a researcher at the Polytechnic University of Marche in Ancona, Italy. And there I started my study. I got my bachelor and master in marine biology in this same university. And then I moved to Spain to get my PhD with Sergi. That was my supervisor together with Andrea Gori. Then I came back to Italy and I started my first postdoc at the University of Genova. And then since 2020, I moved back to the Polytechnic University of Ancona, and now I'm there as a postdoc. Yeah, my main research interests are related with the ecology of benthic suspension feeders. I'm particularly interested in trophic and most of all reproductive strategies of benthic suspension feeders. And just before I came here and I left Italy to come here, I was studying the reproductive behavior of a common Mediterranean gorgonian, which is Leptogorgia sarmentosa. So I was describing the spawning and the fertilization up to the formation of the larvae. So Sergi, don't cry. I know you are really keen to this species. I am a scientific diver, and recently I also got the certification of technical diver, which finally allow me to see directly the species that I only seen by ROV images. So it was a really touching moment for me. And I have to say that all the results that I'm going to present today have been obtained thanks to the collaboration with a technical diving center in Tuscany, in Italy. And they help us with the field activities. So I really hope that I will be able soon to do this kind of work by myself. Okay, so I know you already heard many times the definition of marine animal forest in these days, but I'm going to be a little bit redundant. So marine animal forests are animal-dominated communities mainly composed by sponges, gorgonians, corals, that create a complex and three-dimensional environment that might boost the biodiversity because it attracts many other organisms that could use the forest, the presence of the forest, to search for food, to take shelter, or to reproduce, to a site for their reproduction. Marine animal forests are composed by organisms that are called ecosystem engineers because with their presence or their action, they can modify the environment. And I'm going to present results both related with the presence of autogenic engineers and allogenic engineers. And the main difference among these two categories is that autogenic engineers modify the environment mainly with their presence. So the morphology, think about an arborescent sponges or gorgonians, the presence of these three-dimensional organisms might modify the environment, creating much more habitat for other organisms to live in. While the allogenic engineers are all these species that might modify the environment through their action. Think about bioeroders, for instance, sponges, polychaetes, or bivalves. Road create cavities and inside the substrate, and these cavities, newly formed cavities, might be used by other organisms as an habitat. So a little bit of history. I know that it has been mentioned during these days, but I collect some papers that were published before the definition of marine animal forest was created, was coined. And this list is not meant to be exhaustive, but already in 2009, this publication highlighted that the presence of black coral meadows, cold water coral oases, or more generally biological structure, the presence of biological structure might increase the biodiversity. Not just corals or gorgonians, also some papers take into account the relation among the presence of sponge gardens and the increase of biodiversity. And we have to go back to the 2012 when the first definition and the concept of animal forest was first mentioned in this publication by Rossi et al. And then finally, in 2013, it was described the threat, all the potential impact that might threaten these communities. The real definition, let's say, of marine animal forest was then compiled in this huge effort of many researchers that work with animal forest. And these two books were published in 2017 and 2021. Okay, so animal forests can increase biodiversity, okay, and for the reason they are also considered and defined as essential fish habitat. And with this definition, we mean all the areas and sites that might be used by fishes, but also by invertebrates to complete delicate life stages, such as, for instance, the reproduction. And before the definition of MAF, already this publication highlighted how the presence of deep water coral can be used as a nursery area for fish larvae. And in this same picture, you can see how the eggs of a squid were laid on the top of this big Mediterranean hydrozoan, which is Leptocarpia myriophyllum. So since many fishes and many other organisms use these areas as reproductive site, and so the biodiversity in these areas is really high, these areas are also vulnerable ecosystems, vulnerable to many direct and indirect anthropogenic impacts. And I'm going to present some of the threats that you can find in the Mediterranean Sea, but in the first case I'm going to show you now, the presence of abandoned, lost, or otherwise discarded fishing gears is a problem spread all over the world. So in this case, you see in this video, this is a shallow shoal near the Civitavecchia Harbor, near Rome. This was a shoal covered by a small population of red coral. And the presence of this quite big net abandoned there caused the breakage, the direct breakage of the red coral. And also the problem with these abandoned gears is that the first impact might be the direct breakage, or the colonies, depending on the species, might be bent by the weight of the net. But then the net itself will be covered by the other organisms, and the weight will be much, much higher. And so the colonies that live below the net might directly be suffocated by the presence of the net, or the tissue might be damaged. So now this net was removed, but we already observed some clear damages to the colonies that live there. Another impact in the Mediterranean Sea, is related. This video is not working. Let's try like this. Yes. So another indirect impact of the anthropogenic activities is related with the climate change and the increase of temperature. This is a wall, this is a video recorded in the Portofino MPA. We are in the northwestern Mediterranean Sea, and we are around 22 meter depth. So you can see that the shallowest colonies of this red gorgonian Paramorica clavata are completely dead, and you have to go deeper to see some healthy colonies. The same wall was monitored by us at the beginning of the year in January, and all the colonies were healthy at the time. So it was this impact, this extended death and mortality was directly related with the increase of temperature and heat waves that we experienced in that year. Okay. Another impact indirectly related with human activities and with climate change is the mucilage outbreaks that is much more common now in the Mediterranean Sea. This video again was recorded in the northwestern Mediterranean Sea, around 16 meter depth, and you can see that all the sea bottom is completely covered by this blanket of mucilage. Mucilage is a natural phenomenon. It's a spread of filamentous algae, But the problem is that with increase of temperature and this high temperature that lasts longer, also these mucilage last longer. So all the organisms that live below this mucilage have been suffocated or can die because they cannot directly feed, or even the reproduction might be somehow affected. And here you can see an example. Since these mucilage outbreaks occur usually in summer, which is also the period of reproduction of many corals and gorgonians in the Mediterranean Sea, you can see here how the larvae of this, again, of Paramuricia clavata, the red gorgonian, have been trapped in this mucilage cover. So these larvae cannot find for a substrate to settle and will die because the living period of a larvae is limited. And so all the recruitment for that year is affected by the presence of mucilage. Okay, so all these threats that I listed highlight the vulnerability of the marine animal forest, and I think that you all agree with the fact that the marine animal forests are important habitat for many other associated species that are able to increase the biodiversity. But as Lorenzo said yesterday, I mean, we really would like to see how the presence of this Mediterranean, in my case, Mediterranean marine animal forest, might influence the presence and the living histories of other associated species. So we would like to find a way to directly witness, to directly see how this interaction among species occur, and to have data for us to describe these dynamics, but also to provide data to managers and to conservationists and policymakers to justify and sustain conservation and protection measures. So the method that we decide to use to study the interaction among habitat formers that are the organisms that constitute the forest and the associated species was the time lapse. So we deploy in several locations in Italy this instrument that you see here, that we friendly call Frankie, because it's like a Frankenstein of many different, let's say, instruments that you can follow, that you can easily find in the market. So this time lapse was customized for us, and it is composed by a GoPro 8 in a stainless steel waterproof case that is this one, this part. Then there is this flexible arm that brings the strobe and the light. And then there is this tripod to fix the system, the time lapse to the sea bottom. And then there is this battery, which could be of different power, let's say. We use 25 and 50 ampere batteries, which allow us to record for respectively 6 days and 10 days of recording. And this system is set to take a picture every 3 minutes. And this can be modified depending on your needs. And I say this because it is not a continuous recording. It's not a video. It's a continuous, it's a series of pictures taken every 3 minutes. And then the characteristic of this time lapse is that it could be deployed up to 100 meter depth. But in the sites that you see here in the map, we use it in a depth range from 7 up to 72 meter depth. So the shallowest depth range was 7 in Ancona, near my university. Then we have 2 studies at 72, one in the 2 studies in the Tuscany archipelago, And in this one is a 72-meter depth in the Giannutri Island. And this one is 72-meter depth in Pentapalunno Bank near Naples. The target species that we would like to investigate in the Pentapalunno Bank was the Dendrophila ramea. In Ancona was not directly a species, it was a wall covered by this hole created by two bivalve species that are bioeroders. And we would like to study how these holes are used by other organisms. And in the Tuscany archipelago, we deployed two times the time lapse, one to target Antipatella supinata at 72-meter depth, and one time to target a forest of Paramuricia clavata at around 40-meter depth. Okay, I will start with the first results related with the presence of Dendrophila ramea. To be completely honest, the focus of this study was not to study the associated fauna, but was to study the activity rhythm of the polyps of this colony. But as a side observation, we also observe some dynamics with a sea urchin that we will see later. So to investigate if there was a pattern in the opening and closure of the polyps, we divided the 6 days of recording that our time-lapse obtained in 6 different time slots, all of 4 hours each, following the methodology of Enrichetti et al. 2022. And what we saw is that the majority of the polyps are open during all the time slot, and there is a slight trend of decreasing in the opening of these polyps during the evening time slot. Although we did not see visually anything in the surrounding condition that might trigger disclosure, we found a significant statistical difference for disclosure. So probably there was something that triggered and the closure of this polyp, and we also try to relate this with the decrease of the tide intensity. While we were observing the picture retrieved by the time lapse, we made this interesting observation. So you can see here the presence of this sea urchin, Centrostephanus longispinus, which every night spend and every day spend half of the day resting in either this site B or site A, and at a precise time of the day, which is 8 p.m., it departed from the resting site. He moved up here or in this direction, probably to graze. And it is incredible to see that he came back exactly at the same time. So every night he came back at 4 in the morning. So it was extremely show high fidelity for the site, because it always choose this or this site. And also it was extremely punctual. I don't know the punctual. And he came back exactly at the same time. So we are also sure that it was exactly the same individual performing this behavior, because one of the spine brings some filamentous stuff on it. So we are sure that it was exactly the same individual using the same spot every night. Moving to the next results. Here, the target was this black coral, this common black coral in the Mediterranean Sea, which is a mesophotic species, and the name is Antipatella supinata. And this study was the one that gave us much more data, because we used the bigger battery. So we have 10 entire days of recording in this case. And we observe several peculiar behavior of many associated species. And I will start with the first one. This small fish is a common fish that live in association with gorgonians, with antipatarian in this case. The name is Anthias anthias. And we observe this fish resting every night in the same spot for the 10 days of observation. If you take a look here in this particular spot, you will see, yes, you will see the anthias arrive, and you also see the time passing. So it arrives every day around 8 p.m. and it stay there for all the night. And in some occasion, it is triggered to move because some predators approaches, some, for instance, fishy species of Scorpena. And otherwise, it can spend an entire night sleeping or resting there without, if any organisms, you see now, is bothered by this other organism, this is Scorpena notata. I will show the details of this interaction later on. But if there's anything disturbing this fish, it can rest for 10 hours straight. So I think he slept much more than me. So it's a lucky one. But in other occasion, you can see here, this is the night where he slept less hour, for only 4 hours. And these intervals are related with the amount of time it moves, basically. And it changed the position and then it came back to the same spot. Another interesting observation was related to the use of the black coral colony as a foraging area. In this case, we observe this Holoturian species, Holoturia forscali. Again, you will see appearing from here every night, almost every night, not 10 times, but I think, if I correctly remember, 8 times out of 10, 8 nights out of 10, these Holoturian appear. Always at the happy hour time, around 6 in the night, it appears. And it starts making this path around the colony. So always the same path. So it is probably foraging, searching for food. I mean, during the days, you will see some picture later on, this area is completely crowded of other fishes. So there will be a lot of availability of particulate organic matter to eat. And since this is a detritivorous species, it probably takes the chance of this high availability of food. And this is why it came back every night to the same spot. A similar observation, but this time this crab did not come back every night, but like 4 nights out of 10. And you see here, again, here the crab arrived. Herbstia condyliata is the name of this species. And it starts moving around the colony. And sometimes it stops here in the proximity of the Anthias anthias that was sleeping. Poor guy, been bothered by many species. But probably if he found there much more food because of the presence of the other fish. And we cannot be sure that the Holoturian was exactly the same individual because there was not anything that could allow us to recognize that particular individual. But for this crab, the presence of these sponges, of these decorative sponges on the carapace of the crab, allow us to distinguish this particular individual that came back in the same spot during the days. As I said, this methodology also allows us to describe some interspecific interaction. And the first one, you already see some frames, but it is this interaction between Anthias anthias that were resting here and Scorpena notata, the small red scorpionfish that arrive at some point. You will see arriving the Scorpena here and is starting interacting with the Anthias, touching the caudal fin more and more. And so in the end, the Antias decided to leave. But when it was already 7 in the morning, okay? And 7 in the morning is the time in which normally these fish move and move to the water column to search for food. So here I try to reconstruct this interaction more in details, okay? So the scorpena arrive in this time at 5:24 in the morning, and for more than 1 hour, continuous touching and bothering the Antias sleeping there. Until this moment, in these 2 frames, we observe a peculiar behavior of the Antias that decide to confront the scorpena. So instead of position the caudal fin towards the scorpena, he decided to nothing happen. No, too much. Sorry. Ecco. So here you see that they decide to take another position face to face again against the versus the scorpena. And after this moment, this interaction, the Antias left. But as I said, it left at 7:02 in the morning, which is the normal timing for the Antias to leave. Because during dawn, this is the situation around the colony. So all the Antias, but also here you can see another fish, started to create a shoal, an assemblage around the colony. They create a huge group of individuals around the colony. And then during the day, you can see only few individuals because they spend the entire day feeding in the water column and they come back to rest during night. A similar behavior of the Antias of facing directly is, well, not predator in this case, but this intruder, we can say, was observed with the Loturian. So at some point, also the Loturian starting touching the caudal fin of the Antias. And also the Loturian was probably found a lot of food there, so it was feeding, not intentionally bothering the Antias, but at some point the Antias decided to change position and directly confront the Loturian. And in this case, since this interaction, you can see the Loturian is here, although, well, maybe you cannot see, but he's here. And here is the Antias that changed the position. And in this case, the interaction occurred around midnight, and the Antias, after this interaction, take the original position below the colony. So probably the movement observed with the other fish was related with the timing. It was the time for the Antias to leave and search for food. So to try to summarize all this behavior that I describe with the videos, here you can see, again, we divided the days in 6 different time slots, and the night time slot are represented here in blue, and the white part of the graph is of the figure indicate the daily hours. And the bubble plot indicates the different behaviors, and with the light blue is assemblage when we define as assemblage when more of 5 individuals of the same species are recorded in the same frame. Then there is the feeding, which was observed only performed by these fish or fishes of the family Labridae, and it was highly complicated to be spotted, this behavior. And because it is clear that they are feeding only when they are in upward position with the caudal fin up and the head toward the sea bottom searching for food. The assemblage behavior was performed mostly by Antias during morning and daily hours. While it is interesting to see that all the invertebrates that we observed are present only during nighttime. So they are nocturnal, completely nocturnal organisms. In total, we observe frequenting this colony 34 taxa, divided in 15 invertebrates and 19 vertebrates. And you can see here in these heat maps the number of pictures in which these different taxa were recorded during the different time slot. And it is extremely clear that the decapoda appear and are much more, there are much more pictures showing decapoda during night times, while teleostei are present almost during the time slots, although they are much more abundant during the assemblage behavior that was recorded in dawn and morning mostly. And here you can see how the number of taxa increase in the early night, late night, and dawn time slot, because in this moment appear the invertebrates came, so the number of taxa recorded is higher. Since we also observed in this picture the presence of a strong currents in some moment, we would like to understand if the presence of currents might decrease the number of the species associated with the black coral. Because visually, we see a decrease in the number of fish species or invertebrates moving around. So we perform a statistical analysis, we perform a generalized linear model to check if this observation, visual observation, was statistically significant. And it was. When there is the presence of currents, you can see in the next video, you can see only few species. And I invite you to see appearing here in this spot, to check, to focus on this spot of the colony, because at some point, this fish species, La Panella fasciata, appears, and it seems that it tried to search for shelter against the strong currents in the branches of this black coral species. So in general, there is only few species that you can see. There is the Anthias resting here and here, the La Panella starting moving in a synergistic ways together with the branches of the coral. So maybe it is searching for a shelter to resist the strength of the currents. Okay, now I move to the next results. We are still in the Tuscany archipelago. In this case, we are in Punta Ciana, in a coastal site at 40-meter depth. And the positioning of the time lapse in this case allow us to record the reproductive behavior of this pair of Apogon imberbis. This species is the only apogonid species belonging to this family in the Mediterranean Sea. It is a small fish of maximum 30 centimeter of length. And you can see the sexual dimorphisms among female and male. The male present this bigger buccal cavity because it is a paternal mouth brooder. So it incubates the eggs once fertilized in the buccal cavity. So in this case, the time lapse recorded for 6 days, and we were able to describe the encounter, the first encounter of the pair, the formation of the pair, and the ritual, the courtship of this couple of fishes up to the mating event. So here you see in this picture summarizing all the behavior that we observed and that I'm going to describe now in details. I will start with the behavior recorded that are not directly related with the mating and that are performed separately by the female and the male. So the sheltering behavior was the first behavior we observed. Both male and female take shelter in the branches of these red gorgonians. And we describe this behavior when we observe the same fish in the same position for several consecutive photos. And since the fish was not out of focus, we might be quite sure that it was still in the same position without any movement. Then there is the wandering. Wandering is defined as the fish that is moving from one picture to the next. And it was moving also during the shot of the picture because it was, for many times, it was out of focus. And this behavior was performed mostly by the female. Then there is the bottom interaction. Bottom interaction we defined as the fish that touch the substrate, they interact with the substrate with the head downwards and the caudal fins orientated upwards. And finally, there is the patrolling. Patrolling was a behavior observed mostly by the female and with the fish that swimming around the colony, but keeping a fixed orientation. The fish seems to fix a precise portion of the substrate, and this is probably related with the defense mechanisms of the female once the eggs have been laid. Then there are several behaviors that we observe relating with the mating behavior. So we start with the parallel circling. So once the pair formed, they start to move together, maintaining a distance of 20 centimeters, more or less, among the individuals, and they start to move around, making parallel circles around the colony, maintaining this fixed distance. Then there is the crossing of the tail. So they start crossing the caudal fins. And then at some point, both male and female interact together with the substrate, and this is probably the closure of the mating event when the female interact with the bottom to lay the egg, and the male fertilize the egg and take the eggs inside the buccal cavity. So here you see, yes, the video with the sequence of all these behavior that I just listed. And in the picture, you can see a representation, an easy representation of the behavior we observe. So for the first time, we can be witness of this, of the formation of this pair and all the ritual that they perform to reproduce. And it is interesting to say, to observe that this wandering behavior performed by the female that was like checking, sorry, not the wandering, the patrolling, it was controlling the sea bottom, could be related either by a defense of the area, because these fishes might be highly territorial, or it could be that the eggs have been laid on the substrate and not necessarily the males took it in the buccal cavity to incubate them. I mean, all the apogonids described so far are used to, the male of the apogonids described so far are used to incubate the eggs in the buccal cavity. But since we cannot observe directly this moment in which the male take the eggs in his mouth, we can also hypothesize that the eggs were still there and the female was just trying to defend their eggs. And this is like a limit of this methodology. I mean, it is not a continuous recording, so we unfortunately miss some important moment, but still we can have an entire cycle of reproduction. And this picture just summarize the percentage of time the frequency of each behavior performed by either the female or the males. And you can also see that there is the unknown category because in some cases it was difficult to see the mouth of the male and to distinguish the two sexes. So now I move to the last result. And I'm going to present results related with the presence of allogenic engineers. Here we are in Ancona. So 7-meter depth, and this is the visibility that we usually have in the Adriatic Sea. Sometimes you cannot see your hands while you are diving, and sometimes you lost your body more and more times during the dive. But if you are able to really go close to the wall, it is a site with a high level of biodiversity. But you have to be really prepared and you have to look for it. It's not an easy site to do investigation. But still, we positioned Frankie, our time lapse, to study the role of these two bivalve species, which are Pholas dactylus and Lithophaga litophaga. These two species are bioeroders. They create by themselves these cavities to live in. And once the bivalves died, these cavities are left empty. So other organisms might use these same cavities to search food or to take protection, take shelter against the predators, or simply to reproduce and lay the eggs. This is the video that we recorded for 6 days. Here you can see all the cavities, empty cavities, created by these bivalves, mostly by Pholas dactylus in this case. And you can see some organisms moving around. I know that it is difficult from the video, but I'm going to explain in details what was happening during these days of recording. You see here these mussel these gasterops species, Hexaplex trunculus, moving around, and here another one. And in total, in the 6 days of recording, we recorded 34 taxa belonging to 5 different phyla. And these are the taxa that were much more abundant in the days of the recording. I'm going to give you some more details on the species that were much more abundant and common during the recording. And in this case, you see here Hexaplex trunculus that was mostly not moving during the recording, either during night or days. And this is related with the feeding behavior of the species. So these species stay on directly on the top of one of the cavity, which was still occupied by the owner of the cavity, by the Pholas, because these mollusks fed on these bivalves. And the mechanism of feeding is the drilling of the valve of the bivalve. And after the drilling, it starts sucking the tissue of the bivalve. So this takes some time. This is why for most of the frames, this gastropod was not moving. Then there are several crustaceans that we saw. I don't know if you can see here, but there is this juvenile of Galatea. And here you can see the claws outside of the cavities. And we are almost sure that only the juveniles use the cavities and stay in this position to searching for food. The adult of this species normal take refuge in crevices and not in these small cavities. Then there is the presence of this other crustacean, Pilumnus sp, and you can see again here the claws of the crab, and there is here the cavity. So probably this is a male, and inside the cavity there is the female, and these organisms was seen moving and coming back to the same hole for many days. And this is because to reproduce, the male need to bring a gift to the female. So if the female appreciate the gift, then the mating might occur. The female left the male entering the hole, and they start the reproduction. Otherwise, the male is invited to find another gift, bigger food probably, and to come back with a bigger gift or something more appreciated by the female. It could also happen that during this search for the perfect gift, another male reach the same hole, find the female, and reproduce before this male. And here you can see, I know it's difficult to see, but there are two males here fighting for fighting for one cavity, fighting for the female inside the cavities. So we were also able to see with the time lapse this kind of dynamics and competition among males for the same females inside the hole. Finally, many fish species use these cavities as a reproduction site. In the most abundant species was this small blennid, Parablenus roxy, which is, here you can see the head of the fish that is evident outside the cavity. And usually these species are extremely territorial. So if you approach them, if some predators go closer to the hole, they try to defend the hole, because inside the cavities they lay the eggs. And so they try to defend the eggs and also to defend their home, because they, once the predator is gone, they come back exactly to the same cavity. And from the video, this is the most abundant species that we could see, but by other visual census that we performed in the same area, we spotted many other blennids using the cavities as an home, as a refuge. And also other fish species were seen with the time lapse moving around and searching for food inside the hole. In this case, it is Ranus hepatus, which is known to feed on small crustaceans, mysidiaceans that usually are abundant inside these cavities. And finally, I also have to tell you a sad story. I mean, these bivalves are illegally fished in my area in Ancona, unfortunately, but it is an illegal fishing very common in many areas of the Adriatic Sea. The problem is that to fish these bivalves, you need to completely break the substrate to retrieve the bivalves. And so you are not only going to take away the mollusks, but you are only going to destroy the home of many other, of all the organisms that I just listed before. So it is also, these are also species that take longer to grow. So to take, you can easily take a species that has around 50 years. And to do so, you are going to destroy also this incredible three-dimensional environment that they created. So to conclude, thanks to the time lapse, we have the possibility to record several dynamics among species, interspecific relationship among species, and also to see for the first time some strong diel rhythm and circadian rhythm of different organisms. So I think that is a really powerful method to obtain data for us as a researcher to describe this interaction, but also to sustain why these marine animal forests are so important for other organisms and why marine animal forests support the increase of biodiversity. So we can use these images, this picture, to increase the awareness and to educate citizens from, I actually use the same images with 9-year-old children, and they were having fun searching for the species that were hiding inside the branches and moving around the colony. But more important, we can also use this image to justify with managers and policymakers why we need to protect them and why we need to enforce management and protection strategies. So I have to say that this was not a work that I could do alone. So I have to really give a huge thank you to the scientific team of the marine zoology laboratory that collaborate with me to obtain this data, and to the incredible science diving team of the Argentine divers that help us with the positioning and retrieval of the retrieving of the time lapse. So, and I also would like to thank you, all of you, for the attention.
Thank you, Martina. That was really nice. I love it. Indeed, all this behavioral and demonstrating many things. One of the things that for me is more important is to see that this kind of work is, you know, we are always looking at impacts, or we are looking at degradation, we are looking at, you know, the impact of climate change on biogeography, becoming health cycles, which is also doing this work. Okay, and also population structure and so on. But sometimes just putting a camera there, which is very complicated, I know, and looking at the behavior, I mean, that for me, this is, at least for me, this is very new, and I'm studying the marine animal forest for many decades. So this kind of approach is really fantastic and demonstrates many things, and one, that there are assholes everywhere. Because you have these poor anteas that only wanted to sleep, and the others, the scorpina, live there for 1.5 hours there. But what do you want? They only want to sleep. So, questions? You don't have questions about this? Come on, what's really beautiful. So I do have many questions. I will do only a few ones. The first is about the ghost nets. Yes, because it's controversial. What do you think? Because there is people that is in favor and against to move them in some. When is good to move these ghost nets? Where it's not so good? What do you think about that?
Well, I think that the idea is that if the nets was recently lost, so it was not already epibiontated by many other organisms, or even worse, the nets has already started to be a part of the substrate, then it is that you can remove it, and it is beneficial for the organisms living in that particular wall. But if the nets or the lines are already completely entangled with the substrate, and has been overgrown by other organisms, in this case, I am afraid that you are going to damage much more the substrate than deciding to leave the net there and to leave the other organism to colonize the net.
That overgrown, yeah. In fact, one of the videos that he showed some years ago, we found that in these nets, they were completely, let us say, it's not absorbed, but they are completely encrusted in what was 100 meters, 80 meters depth, 60 meters depth in Maledetti. I don't remember well. We will see on Thursday this video, and then it's not good to just take out this net, because it's already, you know, in every single organism say, okay, this is my new substrate. Okay, and they start to colonize, and you see the gorgonians and other organisms going through the net, outside the net, and that was very interesting. The other question was a little bit, so one of the next steps will be probably looking at, you know, other kind of behaviors and so on. But I know that it's expensive, and I know that it's time consuming, but have you think about making some control, in the sense to put the same camera in nowhere, where it is sand bottom or mud bottom or whatever?
We actually did it in the site where we performed the study of Antipatella, so Giannutri Island. We also position a time lapse outside the forest to check for differences in this behavior and the abundance of species living there. I have to be honest, in that particular case, the time lapse recorded just for 2 days instead of 10. So this is something that happened. But yes, we try to make this kind of comparison, because of course, yes, I'm sure that the presence of Antipatella somehow can boost all this interaction. but it has to be proved also without the presence of corals, what could happen. Yes.
Thank you for the nice presentation. I wonder two things. One is how should the place to the film?
The temperature, it was October when we performed the study. So if I don't make a mistake, it should be around 14 degrees Celsius, the temperature. We are below the thermocline, it's almost stable. So should be like 14, 13 degrees. The problem, well, I forgot to tell this detail of the time lapse that the battery was connected with a wetmate connection. So in theory, you can go there and just exchange directly the battery with a new one and continuous recording. But then still we are a 72-meter depth, so it was kind of logistically complex to do it for a monitoring, a longer monitoring. We are going there. This is our idea, but still we are not ready to this longer monitoring. But
So in line with that, because one of the questions is a very technical question. Oh, sorry. It's a technical question related with America's one. So I understand that you choose the elapsed time of 4 minutes because of battery and memory, or?
Because it was a good compromise, yes.
Okay.
With a picture every 3 minutes, it lasts for 10 days
3 minutes.
with a bigger battery. So we have quite a lot of data to
Okay.
can describe a pattern, a cycle, a circadian rhythm. See, it was a good compromise. That was the idea, yes.
A minha pergunta é se, não sei se ficou claro, mas quando fazia as fotografias, a luz ela só ativava no momento da fotografia ou ela ficava sempre ativa?
Sta domandando se quando stava facendo la fotografia si manteneva attivo. Penso che lei non quando fai la fotografia, una fotografia ogni 3 minuti, cioè spiega un po' meglio come facevate le fotografie.
So the systems was completely automatic system, and it was set to switch it on the light just a moment before taking the picture. So first the light, the strobe switched on, then the GoPro switched on, and then it take the picture. Okay, so it was something that happened like in a few seconds. Everything switched on, take a picture, and everything switched off again, and then the same every 3 minutes. Did I answer your question? Okay.
This time, if another individual take place in the river, this kind of data, you know.
I mean, for the fish, it was quite difficult to identify. I mean, all the antias were almost the same size and identical, basically. It is likely that it was the same fish because I did not have time to show you other pictures, but in a side cavities, really close to the spot where the antias was resting, there was another antias that sometimes came out and go back to the same cavity. So probably there is like a separation of places. Some individual decide to be there and they stay there. I cannot tell you if after 10 days, 20 days, they change their habit and they change completely their spots. But I think this is an observation that confirm a previous published paper last year Bo et al. Published a similar observation, so that the antias rest in the same place during different days. So it's something not new, and probably one individual decide one spot and stay there. But of course, we cannot be sure.
Okay, and all of them are so residents in that area, yes? The individuals of that species, all of them live there maybe for more than 10 days, but do you think after this time, another one will replace that habitat or that spot or that cavity?
Well, I think that during their life cycle, the lifetime, if they can stay there and they can use that spot, no need to. I think that they are quite real. Let's say they decide the site and they stay there. And the thing is that they come there only during nighttime due to their feeding behavior. They need to go in the water column to search for food, and this is why you see them only during nighttime, or within a huge assemblage during dawn, when they create these big groups and then move all together up in the water column. So I think it's related with their life cycles.
Thank you.
You're welcome.
Thank you very much, Martina. Now, Lorenzo wants to show us a video, which a very nice video indeed, about science applied to restoration, okay, with one single species, Corallium rugurum, that is one of the most emblematic species in the Mediterranean Sea and all over the world because of several things. Yes, but before he wanted to show you the video that I wanted to show yesterday is 1 minute video.
So yes, so the first one is because yesterday we were speaking about different forests, and someone came out with the comment that if you go, you can have different forests of different trees, and if it happens also in the sea, and we have a 1.5 minute of that, and then just there was time yesterday came out the comments on the role of the scientists, the way to approach things, and I think that this documentary that we did on a project on red coral restoration is a little bit this. So when you approach to something like restoration or any study, the way going deep, the posing question, and not doing simply a transplant or whatever is important. So we try to show in the documentary, it's 15 minutes documentary. Actually, it's beautiful to see because it's done by one of the most famous video operator that you can find, Roberto Rinaldi. So the image are very good. But the thing is that it's something that show the approach of putting science in first place. And the documentary been accepted to a concourse in France, so will be broadcasted next week. I will not be there, but it's an important film exposition. It's ready. So the first. The first one, the bosque. So the first one is we did it during the Beach Arm project and the Deep Life project in Canary Island. We were focusing on the Antipatella Walla Stony Forest, which is a black coral with colonies that are as high as 1.5 meters. But then when we started to go deeper to find, when I was speaking about finding new forest and pushing forward the limit of the forest, we started to find, and you will see in the video, I will comment why it is going, that we change, the habitat change and the type of forest change with that. Also, these are El sonido también, ¿correcto? El audio. Okay, so here we are in Canary Island. And you see that we can start again. Puedes recomenzar desde cero. No, no sé. No, we cannot see. It's not. Was a good shot. Interesting papel electronic between 50 and 70 meters, and we are focusing on that because it's easier to know the microclimate inside the big forest. That one will go a little bit down. Found the transition zone, which we find also sponges. So there's no more domination of the antipatellar sponge. No, that's not visible. No, it's, you cannot see. But let's see a video, you see some pictures. It's difficult it's difficult to comment, is to scatter. You see changing the forest, but here you can see only some frames. Sometimes it's no word to see it. I think if it's the same with the other documentary, it's not possible to watch. We can try. The other is worse because it's like 4 giga. So this is this 15 minutes documentary is put together 3 small documentary. So you will see that there is the structure made in 3 phases, and this is the follow-up of a project that was funded by the Fondación Transdemocracia.
Le corail rouge est une espèce emblématique de notre mer. Éclatant de rouge, solide, exploitable, il a été pendant des siècles l'objet de chalutage, puis du braconnage et des effets du changement climatique.
Malgré les nombreuses mesures de gestion appliquées à niveau de l'interbassine méditerranée, avec l'objectif de réduire le déclin de ces populations, la croissance lente de cette espèce nous suggère l'utilité des mesures de restauration active, surtout dans les zones où la pression de la pêche a été éliminée, comme par exemple les aires marines protégées.
La restauration des écosystèmes est une branche de l'écologie de la conservation. C'est une science jeune, particulièrement pour le milieu marin, qui concerne surtout les récifs coralliens. Mais ici à Monaco, nous nous sommes intéressés à la culture du corail rouge depuis plus de 30 ans. En effet, c'est dans les années 1980 que nous avons imaginé la construction de grottes en béton de 13 mètres cubes afin de pouvoir faire vivre, croître et se reproduire le corail rouge avant de le réintroduire dans le milieu naturel.
Dans les années 80, j'étais une étudiante de biologie et j'ai creusé les travaux et les recherches de Denis. 30 années plus tard, j'ai l'occasion de donner ma contribution. Sur la base des grottes de Denis, j'ai conclu un système, un véritable laboratoire sous-marin où les colonies de corail rouge pouvaient être étudiées et élevées dans des conditions naturelles.
Aujourd'hui, l'objectif des collaborateurs de ce projet, le Centre scientifique de Monaco, le CNRS, l'Observatoire de Banyuls, supporté par nos sponsors, la Maison Chanel et la Fondation Prince Albert II, est encore plus ambitieux. C'est d'essayer de mettre en place une reproduction contrôlée du corail rouge afin d'augmenter son succès reproductif.
Les coraux se reproduisent par l'émission de larves. Ces larves, d'environ 1 mm de long, sont libérées une fois par an pendant la période estivale et voyagent, transportées par le courant, pendant environ 20 jours. Ensuite, elles se posent sur un fond adapté, se transforment en un petit polype de corail et à partir de ce moment, pendant une centaine d'années suivantes, elles vivront attachées au substrat.
C'est une bestiole bizarre, le corail. Pendant les premiers 20 jours de sa vie, ils vivent dans les courantes marines, dans la colonne d'eau transportée par la courante marine. Et après, pendant les suivantes centaines d'années, ils vivent fixes au substrat.
Les premiers 20 jours sont très dangereux. Les minuscules larves sont des proies recherchées par des centaines d'espèces de poissons. Et en fait, seule une toute petite proportion survit. La plupart meurent avant même de pouvoir devenir un corail.
Voici l'intuition qui a dirigé notre projet. L'idée, c'était de réduire la mortalité drastique des larves qui sont émises dans le milieu extérieur, proche de 100%, afin d'augmenter le succès de la reproduction. À cette fin, les grottes ont été conçues de manière à ce que les larves produites à l'intérieur de la grotte restent piégées dans la cavité, afin de réduire la prédation sur celle-ci et d'augmenter ainsi fortement la capacité de reproduction du corail rouge.
Une deuxième stratégie, en cours de vérification, c'est basée sur une caractéristique péculière de ce nouvel système de grottes, où les colonies peuvent être extraites et réinsérées facilement. De cette façon, les mères en train de libérer des larves peuvent être installées dans un aquarium et aller élevées dans les meilleures conditions.
Les bébés coraux qui sont nés en aquarium sont maintenus dans des situations de sécurité jusqu'à l'âge de 2 ans, quand ils arrivent à former un squelette calcaire. À ce moment, on les transplante en mer, soit sur un substrat rocheux naturel, artificiel, qu'on utilise pour nos études et nos recherches.
Un jardin de corail qui ne provient pas du prélèvement dans la nature de certaines colonies, qui sont ensuite réintroduites ailleurs, mais d'un véritable élevage de corail. Où les chercheurs tentent de percer le mystère de la reproduction et d'élever les larves jusqu'à ce qu'elles se transforment en individus fixés sur un substrat réintroduisible dans les fonds marins. Dans le domaine scientifique, des dizaines d'expériences de prélèvement de branches de coraux dans la nature et de réimplantation dans des zones où ces espèces étaient en difficulté ont été réalisées. Beaucoup n'ont pas pris en compte les problèmes fondamentaux, compromettant ainsi complètement le résultat escompté. Le premier d'entre eux est la compréhension de la biologie reproductive de l'espèce.
Dans le corail rouge, il y a des mâles et des femelles. Et la détermination du sexe est fondamentale pour les plans d'élevage. Imagine une ferme où tu veux élever des vaches et tu ne sais pas la différence entre vaches et taureaux. Ça va être une catastrophe.
Pour cette nouvelle expérience, en plus de continuer à capter les larves à l'intérieur de la grotte, nous avons déterminé le sexe de chacune des colonies afin de calculer quelle était la meilleure combinaison de mâles et de femelles à l'intérieur de la grotte. Et au-delà du rapport mâle et femelle, nous cherchons également à comprendre quelle est la distance à laquelle les mâles sont capables de féconder les colonies femelles. Et le deuxième était quel est le nombre de larves que peut émettre une femelle.
Les réponses à ces questions viennent de la génétique, qui est une science moderne sur laquelle s'appuie une grande partie de l'intérêt de notre projet. Des informations fondamentales sont contenues dans l'ADN, comme par exemple le sexe du coraux, ou sinon aussi le degré de parenté entre le bébé et les parents coraux. Les laboratoires équipés et les aquariums sont des véritables services de maternité où les coraux sont transportés des grottes artificielles en mer jusqu'aux aquariums où ils peuvent se reproduire et pondre des larves qui sont suivies jusqu'à la métamorphose et à la fixation sur des substrats qui sont ensuite transportés de nouveau en mer, en milieu naturel ou de nouveau dans les grottes artificielles.
Aujourd'hui, le centre scientifique de Monaco est devenu spécialisé dans le contrôle de la reproduction du corail rouge et dans l'étude du développement depuis le stade larvaire jusqu'au stade polype. Et ceci pour assurer un meilleur succès reproductif. Pour cela, les chercheurs du centre scientifique de Monaco ont établi des stratégies et aidés des outils de pointe, espèrent aboutir à des résultats qui sont fabuleux.
Pour notre projet, on a besoin des exemplaires de coraux, mais il est un dommage de les extirper dans son environnement naturel. Du coup, on se trouve souvent en face d'un dilemme. Mais c'est la mer, par défaut, qui nous offre des solutions inespérées. Et nageant dans les eaux des bagnoles, sous le fond des bagnoles sur mer, je trouve des colonies de corail rouge détachées, destinées à mourir se lasser sous le fond. Ces colonies sont pour nous une excellente occasion d'étude et une source de larves vers notre élevage. Mais comme ces branches sont tombées, qui l'a cassé dans une des réserves marines plus contrôlées de France? C'est pas que l'homme qui peut casser les colonies de coraux. Ici, un possible responsable. Aussi, un mérou qui rentre dans son trou peut être le coupable.
L'hôtel Corail, ainsi a été surnommé le système de grottes artificielles, est un véritable laboratoire sous-marin axé sur l'étude des stratégies de reproduction du corail rouge et sur le développement d'un protocole de réintroduction dans la nature basé sur les caractéristiques biologiques de l'espèce. Conformément au principe des solutions basées sur la nature, nous orientons toutes nos actions dans le sens de ce qui se passe normalement dans la nature.
Observer la morphologie des fonds marins, comprendre con quels critères les colonies se distribuent dans l'espace, comprendre l'alternance entre lumière et sombre, comment utilisent les courantes. C'est le travail des naturalistes qui nous inspire pour la conception des systèmes de laboratoire sous-marin. Cette grosse sous-marine au fond de sable, avec la veau riche en coraux rouges, c'est pour moi une inspiration. Je l'ai étudié beaucoup de fois, pendant beaucoup de temps, pour comprendre quelles sont les préférences des larves pour se fixer, quelle est la distribution et la différence de distribution spatiale entre les individus femelles et mâles. L'observation de l'architecture naturelle, c'est l'inspiration pour la conception des grottes artificielles.
Bonjour à tous, on est là pour présenter les résultats de l'expérience de coralliculture in situ que nous avons réalisée à Monaco. Et je crois que les résultats ont dépassé toutes nos attentes. Lorenzo, tu peux nous en dire quelques mots?
Ah, si, par rapport à la settlement, les larves qu'on a obtenues se sont fixées sur les substrats qu'on avait préparés sur les aquariums. Et on a vu un taux de survie qui était 10 fois plus grand, supérieur comparé avec les milieux naturels.
En effet, donc en 2023, on a eu plus de 250 larves fixées sur ces substrats, donc des larves nées à Monaco, donc ces grottes expérimentales immergées ici dans les eaux monégasques. Maintenant, il va falloir commencer à étudier, savoir pourquoi ces larves ont choisi certains substrats par rapport à d'autres, et voir comment elles contrôlent leur croissance dans leurs premiers jours de vie fixés.
On a beaucoup à travailler encore.
Tout à fait.
Ces résultats sont essentiels pour l'élaboration des plans de restauration active basés sur des connaissances scientifiques. Mais tous les plans de restauration active, il n'y a pas de sens s'ils ne sont pas précédés d'un plan de restauration passive, c'est-à-dire l'élimination ou la mitigation des conditions qui ont généré les problèmes. C'est comme si on veut planter des arbres dans une forêt qui encore brûle.
Il faut bien avoir en tête que les zones marines protégées jouent un rôle fondamental en fournissant des environnements où les pressions anthropiques sont minimisées et où les conditions écologiques sont favorables à la récupération des espèces.
C'est de là que nous commençons à défendre la mer pour sauver ses habitants. Un univers magnifique et complexe. Un monde extraordinaire, riche en vie. Contrairement à la terre ferme, il est réparti dans les 3 dimensions. Rochers, falaises, grottes, étendues de sable, immenses espaces d'eau libre constituent un monde encore largement inconnu pour nous.
Même imaginer qu'après des années de souffrance, les hôtels à corail mis au point ici à Monaco et à la station de Banus puissent être utilisés de partout en Méditerranée pour repeupler les populations de corail rouge.
You see that what we were speaking about yesterday is that, for example, all these systems started from a question. So we wanted to understand what is the maximum distance of fertilization of the colonies. And I did a joke speaking about the cows, etc. But you know, you don't. Come to contact me that they want to restore coral, and they don't know there is difference in sex, they don't know the details of the reproduction. And in fact, knowing how much is the distance of fertilization is very complicated, because in aquarium you cannot do it because you have big aquarium but not enormous. In the field, and we were speaking yesterday about the question on the sound. You have to control parameters. If you cannot control parameters, you cannot test the hypothesis. So we constructed the artificial population, which each colony was known with the morphological description, genetic fingerprint, etc. And each larva was known the mother, and we find the father with the molecular methods. And from each colony was known the distance from the other colonies. So we controlled all the possible parameters that we wanted to control, and we left the distance and the food, for example, in natural condition, because the aquarium is still difficult to keep completely in natural condition, the coral. And so this is the result of the three-year project, which was the PhD thesis of Bruna, the girl was speaking. And now it's possible that we can see again the other one, the three forests, because there's another, no, not possible. Okay, no, not possible.
Ok, muito bem. Você tem uma pergunta? Sim.
So amazing work, and I would like to ask if there is any other projects about this kind of process with another species.
So in the tropics, there are a lot of projects on coral restoration, and some of them are effective. There's another video that I will show you on tropical corals, and but they are different. What I was speaking about that the Mediterranean, the species are very different. So taxonomically and ecologically and physiologically are different. So we cannot apply the same protocol. So in Mediterranean, as well as I know, there is not a lot of project that was to restoration if he's not putting something somewhere and waiting to see what happens. But I consider it a trials, but not a standardized project with some object. But I don't know. Carlo is doing something with the caves, no? And Cladocora, sì. They are putting Cladocora.
I think amazing project, by the way.
Ok. Un'altra pregunta.
Lorenzo, do you know how long it takes for the red coral to achieve sexual maturity and start brooding?
Probably 6-7 years, around 6 and 10 years. But it's also true that it has sexual maturity, but when it's 7 years, there's like 40-50 polyps with a 0.8 fertility. It means that it does not produce a lot of larvae. But usually 6-7 years should be reproductive. Thank you.
Tomorrow, Dr. Godoy will talk about restoration also. So we'll have the opportunity to make more in-depth, vision of restoration and marine animal forest. Agora vamos todos a comer alguna coisa y voltamos a la 1:30 más o menos aquí para retornar a fazer as presentações. Ok?
Alô, Tarina? Me escuta? Liga teu microfone só para ver se eu te escuto.
Alô?
Ah, pronto, perfeito. Eu resolvi fazer aqui diferente. Eu fiquei com os dois computadores aqui na minha mesa, aí eu vou botar a apresentação aqui no meu, aí eu vou só testar se ela tá passando dali onde o palestrante vai ficar, tá bom?
Tá bem.
Vou aqui fazer um teste, um minutinho.
Consigo ver.
A apresentação tu vê, né?
Sim.
Tá, eu vou só colocar aqui agora ela ali no projetor.
Tá passado.
Vou parar aqui a apresentação. Agora eu consigo me comunicar contigo e te ouvir aqui no fone. Então ficou melhor.
Sim. Sim, tá bom.
Cancelar. Tá. E ali eu consigo ver o... Perfeito. Pronto, viu? Agora é só aguardar. Eu ainda não estou com a apresentação.
Então tu agora juntaste os dois computadores um ao pé do outro, não é? Estás com os
Isso,
dois
eu
um ao
fiquei
pé.
um do lado do outro, é porque eu estava
Sim.
na dúvida se o passador ia chegar aqui onde eu estou. Aí agora eu estou com os dois computadores e consigo me comunicar contigo com esse que... E o dos palestrantes está aqui comigo. Mas aí eu vou continuar usando o outro computador para passar a apresentação, porque o meu tá... Meu PowerPoint, ele é antigo. E aí eu percebi que ele não vai funcionar com o passador, tá bom? Pronto. Tá ok. Quando o pessoal for iniciar, eu te sinalizo aqui, tá bom?
Ok.
Tá bom, até já.
Já.
Vamos a comenzar. ¿Está todo listo? Ok. Boa tarde a todos y a todas. Vamos a comenzar a las de la tarde. Tenemos 2 seminarios. Después de eso tenemos una pequeña tabla redonda, una pequeña mesa redonda, que hablaremos de otras cosas diferentes de las que hablamos ayer. Vale? Entonces, bueno, vamos a ver. Ahora me tengo que presentar yo mismo. Y soy muy triste. Mas a persona que tenía que hacer de chairman, que es la profesora Elena Cascón, no está porque está un poco enferma con bronquitis. Pues no puede hacer la presentación. Me presento yo mismo muy velozmente. Yo soy Sergio Rossi, soy profesor aquí titular de LABOMAR, de la Universidad Federal de Ceará, también con un contacto directo con Europa a través de la Universidad de Salento, donde soy profesor también. Y, bueno, la mia especialidad es ecología bentónica, ecología trófica de los organismos suspensivos. Hice un doctorado a través de la Universidad de Barcelona, también a graduación. Y, bueno, hice un itinerario de postdocs y de cosas realmente muy, digamos que, elaboradas, vamos a hablar así mismo. Y he tenido el privilegio de venir de hacer muchos stages y hacer muchas campañas fuera del país de origen, que era en España. ¿Vale? Y finalmente pues vine aquí y aquí pues me estabilicé también como profesor. Y bueno, ya está, no hay mucho más que decir de mí mismo, tampoco creo que valga la pena perder mucho tiempo en eso, francamente. O que a mí me gustaría hablar hoy de un tema que es un tema bastante complicado, por decirlo de alguna manera. And I will shift to English now to talk about how to regenerate the functionality of the marine animal forest, which is a hot topic from my point of view. And we will, I will try to give you some tips about what we are working with and what are the targets that may ameliorate this kind of approach in general in the different areas of the world, okay? Trying to be sure that the message is quite clear. And at the end, I will just be a little bit provocative about the things that we are doing that may be interesting and maybe help or maybe a tool to understand how to proceed in the near future. So, why the animal forests are important, or why in general the forest of the sea or the marine animal forest in particular, which are these three-dimensional life structure, these habitats formed basically by methozoans that that may be mixed in the areas in which the light is available, maybe mixed with macroalgae or calcareous or fleshy algae, okay, kelp, or calcareous algae or other kind of organisms. Why they are so important? And they are important because they have or they supply many services, not only to the humans, but in general to the habitat in which they perform and they construct. So they are indeed ecosystem engineers that facilitate the life of other organisms. I'm very happy to see that there's many people in all over the world, and here we have some examples like Lorenzo, and like, for example, people like Martina, like Americo, that are working with this kind of approach in which we try to understand not only the importance of the marine animal forest, but their role, try to demonstrate that this list of ecosystem services or the natural capital which we are talking about is just reinforced by science. Okay, we have the IACTA's barrier for hurricanes and storms, the forests of the Sierra, the key habitat for life stages of many species. They feed and shelter vital species of commercial interest. They transform the hydrodynamics, and so on and so on and so on. This is an important asset that we have to take into account because they are allies. So in some way, they enhance biodiversity. They enhance the possibility to fix carbon. They enhance the biogeochemical cycling and the complexity. The word that perhaps is more important is complexity. Is understand that complexity is, how can I say, is one of the things that you have to take into account when you are talking about forest, terrestrial or marine. Forest means complexity, okay? So what happens is that Among other things, climate change is on the wheels, on the track. We thought that climate change was a train at 18 kilometers hour, and perhaps it's now 160 kilometers hour. So the fast climate change, let us say, in brackets, behavior is just accelerating, and we do have a problem. I don't know if we are here or here. I wouldn't enter in this point, okay? But perhaps most of you have clear where we really are. The thing is that when we are looking to this climate change, we see that there are many stressors, among others, the climate change that are impacting directly the forest of the sea, okay? However, even if we do have several impacts coming from the excess of CO2, methane, and other greenhouses, like, for example, the change in the pattern of circulation currents, the warming and ocean acidification, the deoxygenation, and eutrophication decreases the water transparency. Remember, for example, that the slowing down of the currents are making more difficult for deep sea forests to survive because they have less oxygen coming from the surface. The decrease of organic matter delivery because of the stratification and change in the trophic relationships, the impacts on biodiversity and the nursery effect of all of these related basically to climate change, also the sea level rise and so on. This makes, clear that we do have to understand what happens with these impacts without coming from the climate change. However, even if it's this truth, and this is, I love this drawing from Alberto Gennaris, and Lorenzo talked about it, and also Martina talked about it, okay? The thing that the populations are shifting. The populations are changing their distributions and their survivorship because of climate change. In this case, we do have here, okay, healthy populations. You have a heat wave. The heat wave just starts to impact the shallower populations, which disappear. And I can assure you that it is truth in the sense that when I was making my PhD in the Mediterranean Sea, we looked at this forest of the sea of Paramorithia clavata and so on, and I still remember to find this Paramorithia clavata at 13 meters depth, very healthy, and with a very spread all about the coast of several areas. However, look at what I just asked yesterday. What was the baseline? Because for me, the baseline was that. But for the fishermen, which were just working with red coral, for example, that was not the baseline. They worked in the 1960s and 1970s. And in this time, what they found is that not only the marine animal forests were shallower, but they were bigger. They were huge gorgonians, okay? More than a 1.5 meter height, okay, at 20 meters depth, which they completely disappeared when I just was starting my PhD. So take into account this point always, which is the baseline. It's not so easy. And it's not so, how can I say, it's not so rapid that the conclusion that you have to take into account. No, we can make this. Okay, start with the baseline. And of course, you can be sure is that yes, climate change is guilty. Climate change and ocean acidification is guilty of the disappearance of the marine animal forest. But in this case, guys, this, which is bottom trawling, is much more guilty in this moment than the acidification and warming. Warming and acidification are coming, and this is good, this is fantastic, and we can explain many things. But I can assure you that during the last 150 years, and in some places even more, bottom trawling destroyed completely the forest of the sea, not only the marine animal forest, but also seaweeds and seagrasses in many places. In fact, just as an anecdote, just a snapshot, in the 15th century, 1454 or something like that, there was a plea, the people of the coast in England, they asked to the king to stop bottom trawling. They made bottom trawling by roaring, okay? They stopped bottom trawling because they were destroying the fauna and flora of the area, and then they found that they had less fish. 1456, still we are here, and this is not bad, okay? So, the marine animal forests are under siege, the marine animal forests are under threat. And this, which it was illustrated by Lorenzo the other day, is a fantastic view because, of course, what you see is what you protect. And we are not protecting the marine animal forest nor the vegetal forest. We don't care because it's underwater and we don't see it. That's all. And if you don't see it, it's not part of your life. Indeed, if you find a wildfire, when you find, for example, a destruction because of logging here in the Amazonia, everyone is just, you know what, not everyone, but most of the people is, you know, saying, oh my God, we have to stop this. But these poor guys here, 100, 150 meters depth, 200 meters depth, basically nobody cares. Okay, and this is about this base, these shifting baselines. This is, for example, a work about the deep transformation of intertidal coral reef in Australia. Okay, the first picture is from 1893, and this is, you know, the structure of intertidal coral reef, and this is 1994, so 100 years later. What happened here? Trampling. All the people trampling. And the people trampling and just making a walk there, the people is not aware of what happens. I mean, it's not, you are guilty, you are trampling. No, but things have their consequences. But most important, things have their consequences, and the complexity here is much higher than the complexity here. So this probably, more than probably, or for sure, we just have the possibility to gather more biodiversity, more biomass, and more of course, running well associated carbon that is immobilized. The same about coral, the red coral that was that Lorenzo was talking about just in the video just a few minutes ago. So red coral that we see now has nothing to do with the red coral that was present in the past. And here you have a proof. This is an 18th century, okay? 18th century? I think so. It's in somewhere. I think it's 18th century, not completely sure, but 18th century in Sicily, the southern part of Italy. And what happens here? You see that the red coral is poached, is just harvested by hand. But more importantly, look at the guys that are taking this coral. They have no fins, they have only small goggles, which means that they are going 5, 7, 10 meters depth, maximum. Come on, one. They have no stones, they have nothing. So they just go down and without fins arrive to whatever, 5, 7 meters depth. What's happened here? What is important for me is not only that you see the size of the corals and the depth, you completely transform the baseline. This baseline any longer exists and would never exist again as it was, probably. Probably in 500 years, yes, but okay. So we have to take into account these things to start understanding what we decide and what we want to do. So functional regeneration has to be based in some way, in some tiers, okay? The first of them is this marine forest species mariculture, which is marine forest species, okay, for ecosystem restoration, understanding the organisms, understanding what you're talking about, understanding the life cycles, understanding the trophic needs, understanding the interaction with other species. Because if you want to make silviculture, and you want to make silviculture terrestrial silviculture, you cannot put together determinate kind of trees, determinate kind of herbs, determinate kind of shrubs, or determinate kind of vegetables. So when you're making silviculture, you have to understand what you are doing underwater. And if you want to make complex silviculture in the habitats, you have to understand what you are doing. The same is the ecological engineer. So the ecosystem ecological engineer properties, okay, and the applications. So this kind of tools that we are proving to try to understand which of them are better, which of them are the most optimized, which of them can be indeed applied. Again, I would say here brackets. The problem is the scale, temporal and spatial scale, guys, because this is fantastic and maybe good, okay, but you have to be realistic, okay? We will talk about it in just a few minutes.
Then the adaptation based on marine restoration forest. So proof of concept and advanced stage. So try to make this kind of experiments in which you try to understand if the animal forest that you are, or the vegetal forest, because this is also applied for seaweeds and seagrasses on mixed organisms, that they can be perfectly compatible. Okay, for example, calcareous algae and gorgonians, calcareous algae and, for example, ascidians or sponges. When you make this, be sure that you make proof of concept robust enough with statistical models that indeed demonstrate that this may be good, and always tailor it. What does this mean? It means basically that are adapted to the specific area in which you are working. Okay, this is why we try to understand the different ecosystems, and we lack a lot of information here too. Ecosystem seascape, not yet discussed. Nobody discussed about the seascape. So you have the landscape and the performance on the landscape, but the seascape, nobody still is working on this kind of concept of what happens with all the communities that are and how they interact. And finally, you understand what is the transformation of the ecosystem services derived from all these points of detail. So I will pay attention only in one thing to just understand, because it's completely impossible to make or to just focus your attention in everything. There are many groups that are working on that. And in my case, on the groups in which I'm working with, we try to understand, okay, the trophic ecology. So which is the rule of the trophic ecology in this system, how this really works. So remember one thing, here you have heterotrophic and autotrophic inputs, okay? All the suspension feeding organisms in the marine animal forest, all of them need heterotrophic inputs. Even the best coral that you have, for example, the most performed photobiologically speaking, coral needs some entry of phosphorus, nitrogen, and other elements from the prey, from the ceston, a live plankton or the tritus or whatever they feed. Then you have fully heterotrophic organisms, and you have symbiont organisms in which an important part of the transfer of the energy comes from the symbiosis. On the other side, you have the autotrophic input. The autotrophic input basically is those which is the release is transferred from the symbiotic algae. You know that. I mean, here you have many examples of corals and hydrocorals that are working with this kind of symbiosis in which you have this autotrophic input. And then, that's very important, you have to balance these energy inputs with the energy outputs, respiration, growth, excretion, reproduction, defense, secondary metabolites, and so on. So, when you think about whatever, conservation or restoration, rewilding, that we will discuss about it a little bit later, you have to balance both things. You have to take into account what happens here and what happens here, okay? So if this is deficitary, more than probably this will be deficitary. And then remember one thing, the first thing that you stop more or less will be probably the growth, will be probably the reproduction. You cannot stop respiration because if you don't respire, you die. But the other ones, you have to stop. I give to you an example that is very cruel and but it's very real. When the woman going out from Auschwitz, okay, in the Nazi camps, they were, outside that they were fertile because they were 14, 16, 20 years old. Most of them lost completely the possibility to reproduce because all the effort was centered in respiration and survival. All the other mechanisms were stopped. Yeah, I know that this is a very, stressful thing, but this clarifies you what is really the rule of nature. Nature tends to make you survive. And if you survive, then you have the chance to reproduce, which are both things that are the only target of nature in whatever living organism. Survive and reproduce. All the other things are peripheral. Okay? So in these energy outputs, this has to be balanced. And then we have to take into account this kind of entries to understand what is happening and what could we do in the near future if we want to make some kind of activity of conservation or restoration. So what do we have about heterotrophy? These organisms are machines of capturing particles in the heterotrophic point of view. They just extend their polyps, they open their oscula, they open their valves, and they feed in different sizes and at different rates. And they are, very efficient depending on the structure, depending on the position on the toward the currents, depending on many factors that make them more or less efficient, okay, in this way. So there is room for everybody here. There are lots of organisms that can live from the heterotrophic point of view because of this kind of adaptations, of these heterotrophic adaptations. And in fact, one thing that you have to take into account is that when we're talking about the carbon that is flowing in these organisms, we have to take into account the origin of this carbon and the fate of this carbon. Here what you have is basically an algal bloom in Antarctica, okay? That may go from 100, more or less here, to 500 tons per kilometer square per year. It's a huge quantity of carbon. Part of this carbon is eaten by benthos, so the benthos, bryozoans, gorgonians, can eat this directly or indirectly. So directly, feeding on phytoplankton, indirectly feeding the copepods, euphausids, and other metazoans or protozoans and bacteria that are just living in this kind of water column and preying on that. They capture part of this, and this is partly transformed in benthic growth. Remember the scale, okay? The benthic growth is the growth of the organism. So in 1 year, one acropora, for example, cervicornis, one branch can grow 6, 7, 9 centimeters, only one. Instead, in Antarctica, the organisms, for example, grow very slowly, most of them. Okay, then part of the carbon is immobilized. Immobilized, what does it mean? It's immobilized in the structures of the organism. The branches, the massive bodies, the sheets of the organism which are there spread in bidimensional structures and so on. And only a very, 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 very low quantity is sequestered zinc. So it's permanently sequestered in sediments or in other places, which is so this is about heterotrophy. And what about autotrophy? Well, these guys, these small guys here, are providers of food, are providers of translocating the fatty acids, translocating the sugars, and translocating, for example, the amino acids, are capable to maintain the corals. You know exactly how it works. These guys basically make the photosynthesis and then translocate in terms of organic matter that helps in some way, not only for the respiration, but also for the growth of the tissue and also the calcification. So now we have heterotrophy, autotrophy. These are the two things that we have to understand to better comprehend, to better be sure that we have the tools to understand the possibilities of these organisms in front, for example, of climate change, on warming waters, or slowing down currents, on the transformation of the primary productivity, and so on and so on. So, to resume this, we have organisms that are completely here, heterotrophic, for example, deep reef, deep coral reefs, polar sponge beds, oyster reefs, and the organisms, for example, like tropical reefs, tropical Gorgonian forests, which depend much more about the light and about the photosynthesis than the heterotrophic input. Depending on the habitat, you have one or another. And then we have here the outliers, poor guys, the chemosynthesis, the hydrothermal vents, and the hydrocarbon ships. Okay, these guys are just here, just making their own job, but at thousands of meters depth. Okay, so we have to pay attention about this gradient and about the needs of every single species to better understand who will survive and who will not survive, depending on what? Depending on why what is changing in the water column, both from the heterotrophic point of view in explanation, all the things related with the primary productivity and secondary productivity and microbial loop, and second, about the autotrophic input, which is basically not the light that is arriving, which, okay, may change slightly, but the temperature that is related to the photosynthesis. Remember that if we increase the temperature, the photosynthesis just starts to go crazy, and at a certain moment, it becomes toxic for the animal, for the microalgae. So there is bleaching, for example. So the performance of the autotrophy and the performance of the heterotrophy will be very important to understand what we just can do from the conservation point of view or the restoration point of view. So we can make a plan, okay? And what about this plan? What we can do? Well, the first thing that we can do is just understand the impact on the water columns of climate change in the primary and secondary productivity and the biogeochemical cycles associated. That's not easy at all, but there are many things and many approaches that can be done. For example, understanding first the impact of climate change in phytoplankton, making experiments with phytoplankton, coccolithophores, diatoms, dinoflagellates, and making this possible to understand who will be prevalent. Making this who is prevalent, then you can understand also what will be the concentration of food in these organisms, in this phytoplankton, and how much of this food will be transferred to the secondary productivity. So you will have the performance of the phytoplankton, which will be impacting the performance of your suspension feeding organism. The more the food, the more the performance. The less the food, the less the performance, okay? Same thing with the photosynthesis, okay? Here you can have in this kind of approach what will happen in different conditions of temperature and acidification. Then, understanding the food web shifts. Trophic webs are reshaped, less food for baby suspension feeders. As I told you, the impact on the water column, the impact on the productivity and on the carbon fluxes and nitrogen fluxes and other element fluxes will directly impact these organisms. And then, understand the circulation and conductivity changes, so the transformation in dispersal properties, the effects of recruitment processes, and changes in population and community stability. So understanding, for example, one thing, imagine that you have less primary productivity, and this primary productivity is having a repercussion on the quantity and quality of food that is arriving, for example, to a bivalve, or is arriving, for example, to an ascidian. This will have a repercussion, as I told you, in the outputs, and possibly it may have repercussions in the number of larvae that can be released and the performance of those larvae. And as was in the video made by Lorenzo and his team, they try to make you understand that the performance of the larvae is essential to understand if they can survive or not, because they would, for example, they can be less time swimming, less time trying to find home, less time to just settle down, and especially also they may be weaker in front of the organisms that will be competing for space. Then we can understand the carbon immobilization, cis-kept losing efficiency, carbon sequestration, and oceans climate regulation compromises. So one of the parts of the alliance that they have these organisms is that they may just immobilize carbon. As said, for example, Professor Ernesto, it's truth. I mean, these guys are not saving the world. We don't pretend to make restoration, to make these organisms make the mess that we have made with these 420 ppms. No, of course not. It's not. This is not on the mind of a serious scientist, whatever he or she is. But it's true that they act as allies because they perform complexity. And in some way, they can enhance biodiversity, making more and more forests that can gather more biomass, can gather more carbon, and that can be a good ally for the natural capital. And of course, finally, biodiversity at risk, the change in the species distributions, the community structures and ecosystem services are at stake. So they will change depending on all this. So we may have here a plan. And then it comes this two very horrible drawings that I have to stop a little bit here, because this is what we are working with. This is a paper published last year, 2024, in Biological Reviews, in which a group of scientists all over the world, thanks to the cost action, okay, we were starting looking at the gaps of knowledge that we have in heterotrophy in this case, basically centered in heterotrophy, so the carbon flux that is coming from capturing particles, whatever they are, bivalves, ascidians, sponges, and so on and so on. So the first that you have to take into account is that you go from an organism to the ecosystem. These steps are very difficult. So this paper relates you one thing. We still have a lot of information missing, a lot. Because even the more general concepts and the more general approaches may be not well adapted to many habitats, many organisms, many populations. In fact, for example, let me give you some example here, okay? When you look, for example, okay, state of knowledge of a population, okay, interspecific difference delineate trophic niches, yes. Niche partitioning and contrasting heterotrophic abilities, yes, we do have this state of knowledge. The heterotrophy is important during environmental stress. Yes, we do have information about that. But look at this, causes and consequences on intraspecific variation. So what will happen if in the different populations, if, depending on where they are just positioned? For example, many years ago, I had the opportunity to see that in a same wall, in a same Gorgonian population, Gorgonians that were more exposed to the currents were much efficient reproducing. They have much more lipids and they have much more number of gonads per polyp. Indeed, the poor Gorgonians that were in the bottom side, they have less food available, they have less exposition to currents, they were more exposed to resuspension, and then they had the problem inside the same patch to just survive. So this kind of things we have to understand, because then you may have a population that is very healthy at, for example, the northern part of Catalonia in Spain, and the same population, the same species 40 kilometers away in Bagnols-sur-Mer may have less possibilities to feed properly and to perform their reproduction and their survivorship. Then the community, you know, with communities differ in their composition and levels of productivity. Yes, we do know that, even if there are some gaps of knowledge. That, for example, the knowledge of the community about the energies and nutrient fluxes are still scarce. Still, we have a lot to learn about what happens. So, is this disencouraging? Okay, no, if we take this seriously and we understand first the steps and then which are the tools that we have to apply. For example, ingestion rates, gut contents, fatty acids, you know, sterol, stable isotope analysis. Here, you know, demographic traits and analysis. Here, incubation chamber analysis, mesocos experiments. Here, in the case of the ecosystem, particularly organic matter in situ quantification. If we have clear the model and the tools, and if we have clear what we want to respond, and if we have money, this is the most complicated thing, and if we have people that help us, students, PhD students, graduate students, postdocs, and of course, other teams all over the world making similar work to just contrast different habitats, these questions can be answered. And then here about what is the possibility of different actions, okay? Which ones are really, let us say, possible, or we do have the possibility to answer the question, and which ones we do not have this possibility yet. Okay, from organisms again, to the ecosystem. And then we have the actions that we can do, and then which are the risks and limitations. For example, actions. Implement marine protected areas to maintain trophic interactions that ultimately enhance MAF heterotrophy and promote their connectivity. Redness, yes, because we do have these marine protected areas in many parts of the world, and there is people that has been working in the trophic ecology during several decades. So we can say that, for example, in conservation, we have this ready. And also, if we want to make restoration, we can learn about this here in these areas. Okay, which are the risks? Known, in the sense that the only risk is a political risk or a monetary risk if the marine protected area is not financed. Let's be clear about that. Okay, and then the limitations, often designed based on past and present or present information without consideration future conditions, is truth. So when we made a marine protected area in 1970 or 1960 or 1980, we were not thinking about climate change, and we were not thinking about connectivity. We were not thinking about the possibility that this kind of communities were deeply damaged, for example, for this problem. Preserve ecosystems with high food supply to marine animal forest organisms. Okay, climate refuges. So make this kind of preservation of several areas that we identify that are healthy. Well, yeah. The risk is protect only a subset of regional biodiversity, are a solution that depends on local decisions. And the limitation, okay, rule of as a refugee can vary in space and time. It can also depend on its connection to network of marine protected areas, never been applied. So if you see many of the things that you have here, I wouldn't make the check of every single box. It's completely, you know, it's time consuming. I don't think we do have this time. The thing that I wanted to just to show you, and I will show you one of the ones that is in the red area, okay, is that most of them depend on what? Financing and politics, laws, surveillance. So the direct human intervention. So we do have this problem, and this problem is real. Here, for example, okay, macrobiome manipulation of the holobiont, readiness, note, and why not? Well, because we don't have any clue about the macrobiome of gorgonians, coral, sponges. We still don't have practically anything about the macrobiome. We start now to understand what is the macrobiome in this organism. So the possibility to apply this for the moment is not ready. And then R, which is the risk, decrease in genetic diversity of the microbiome. Of course, because when you are manipulating things, whatever they are, you are decreasing the possibility to have more variability within the same population or in the same organism. So here is one real problem that we may have, as well as the assisted evolution. The assisted evolution, depending on what you are doing, we may have this risk. And as yesterday Lorenzo said, when people is thinking about, you know, performing a species, like for example, shepherd dog, the shepherd dog, German shepherd. So when they made that, they were not thinking they may have a problem. They just make them because they need this kind of dog to make, to have some functionalities. But later on, they understood that this may be a backfire. And then the limitations, unknown if the microbiome affects heterotrophic capacity. Yeah, in this sense, I give just a few examples, these examples, and then I go on with that. So what I wanted to show you is that you have to base on that and many other things to make conservation and restoration, regeneration, rewilding plans. If not, it's more than possible that you wouldn't be successful. And we don't have time, in the sense that we do have to understand things to make as much as we can, because it will be impossible to make any kind of thing perfect. Proofs of concept of these proofs of concept that can be upscaled and can be realistic. And if not, possibly you say, guys, I tried, but it's not realistic. This wouldn't work, or this is not feasible or possible. So this is Ocean Citizen, okay? This is the project Ocean Citizen that we are doing in 5 different places in Europe. One is the Arctic, the other is the Baltic, the other is the Mediterranean Sea, the Red Sea, and the Atlantic. And why we are doing that in 5 different places? Well, because these are 5 different ecozones in which you have very, very different environmental variables. So trying to make the same protocol, or better, to make a similar protocol to evaluate the possibilities to make restoration is a first step. In fact, this is one of the things that we are fixed, that we are just promoting. Making this possible in different areas will make, you will start in some way to understand if it is realistic or not, make this kind of restoration of the capacity and functionality of the system. So here what you have is basically a land area in which you have a place in which you try to transmit the know-how to the people and the awareness on what is happening. Here you have a laboratory where you can just make possible the transplantation and the optimization of the transplantation in situ. Here you have places in which you can have the organisms just as a nursery place, also the floating reefs, and then you have active restoration and passive restoration at different depths. In shallow areas, you can make active restoration. In deeper areas, you more than probably will still make passive restoration, which is adding substrate to the seabed, adding substrate to the soft bottom to just make possible this kind of complexity and enhancement of this complexity, okay? In the meanwhile, you will make the follow-up of everything, of the currents, what happens with temperature, what happens with the nutrients, and so on and so on. Okay, so just to finish, okay, this paper impacted me a lot in the sense that when I read it, I understood that if we are here and if we are in the point that we are, you know, we are in a crossroad in this moment and we have to understand what we can do. And this is, as I said yesterday, complicated, okay? So we must take a decision. We are part of nature. So we are part of nature and we have to understand that as part of nature, we have to react in one way or another. It's a possibility of making nothing. You make only conservation. You conservation the 30% of the coasts and several offshore areas, okay? And then leave the nature going per se. But as far as I understand, the problem is that when you do that, it's okay, it's perfect. Nature has all the time to recover, has billions of years to recover, billions of years. For nature, it's the same if we are or we are not here. That's the point that we have to understand. That's the only thing that we have to understand. For nature, we are nothing. Nature is not at risk, guys. It's not at risk. It's the human being, the human species that may be at risk. Not because there will be a zombie apocalypse or there will, no. Because our life will go to worse and worse and worse, and probably there will be some huge constraints in the near future that maybe we cannot overcome as easy as we think. So one possibility is this. The other possibility is try to take action and say, okay, we will try to make something, for example, you know, regeneration plans in which you try to restore different habitats and enhance biodiversity, complexity, the biogeochemical cycles, and so on and so on, to try to recover this functionality of nature. It's a possibility, always based on science. But this, for me, is the sentence that gives you the possibility to understand how. We have to focus restoration sciences on the long term, centuries to millennia, reassembly of the great ecosystem complexity, integrated interaction network, and evolutionary potentially approaches. What does it mean? It means that if we make restoration, restoration is not thought to be made for the next 5 years. Sorry about that. You don't have to think about you or me. You don't even have to think about your sons or daughters. You have to think about your nephews. That's the point that we have to understand. So recover the functionality is not focused on us and our success, because you will be successful, that will be fine, and will be perfect. No, it is a very long-term, target. At least from my point of view, this is good. And this is why I'm in some way, prone to this very controversial, because it's controversial in terrestrial areas, and now starts to be a reality on the seas, of the rewilding concept. The rewilding concept is basically leave ample areas which they will self-regenerate, but with the help of some help of human intervention. Let me give you one simple example. A river that is straight, that is just a line, because we make a channel. Then you destroy the channel and you make the river again going as it was just 200 years ago, for example. And then all the functionality of the river completely changes. This is rewilding, but there is human action. There is active human action there. The same, for example, if you put the famous, hopefully you all know that, the famous wolves in Yellowstone. And then you put the wolves in Yellowstone, and the wolves in Yellowstone start to what? To hunt deers. And all these deers, minimized and more controlled in some way, were less impacting the small trees that start to blah, 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 blah. And during the next 20, 30 years, at the end, even beavers and other organisms were just going from nowhere, but there was direct human intervention. With that scientific program followed like that. Not all the rewilding is good. Not all the rewilding is well projected. We are humans. So we will make mistakes, and also it's truth, there will be people that will be, waiting to change nothing for make this kind of actions, that will give, but I make something and I have carbon credits and so on. But you have to think that, is a possibility, and you also have to fight with that. Okay, thank you very much. And if you have questions. I cannot make questions to myself. You don't have questions. If you don't have questions, I have another slide, but it's very provocative.
Thank you very much, Sergio. It was really, really interesting and sort of make us think about a lot of stuff. And ourselves, when we were discussing in Coral Vivo, what's our approach in terms of coral reef restoring South America. So as I told you yesterday, we had this paper, and when we were discussing and changing our own views about this, the BNDES, the federal bank, to just open the call, and part of the call was towards restoration coral reefs. So we thought a lot, and we choose to in our first step towards restoration, to do research on larval enhancement. So as we look all the options that we have in that same menu you place there, a lot of people doing different things, and we were also looking all the mistakes they were doing and things that were not really working, and we thought that this one could really get into something good, but not in terms of really going to, telling people that, oh, we're going to save our corals, just, enhancing larvae everywhere. And that's not the purpose. We are doing research to see if it works. and develop technology in order to maybe get into something that works. So I want to learn about your view of this sort of, because this is not really like listed in your options for restoration. I just want to learn what you think about it.
No, I completely agree with you that we have, that's also that's complicated, because where is the limit now of the scientists? We know that we are in an emergency that we never saw before. Of course, there were other climate changes and there were other problems when there was a deglaciation. There were human populations that were extinct and became extinct because they couldn't adapt and so on. But now we do have the tools and we have a lot of information. So this narrow, very narrow between just making things and then stop and give to the different governmental entities, local, regional, statual, or federal, or is enough, or the scientist has to go beyond. And this is torturing me every day. So I'm serious about that, because I don't know exactly what to do. Because when you enter in the politics, when you enter in the activism, automatically, of course, the system is ready to say, okay, you are an activist. But no, but I made a lot of experiments and I made a lot of science, and no, but you are an activist, and then you take part. And then when you take part, you are part of the problem and you are part of the fight, and you take one side or another, because humans are, you know, making this kind of approach. So I agree with you completely that we do have to give all the tools possible with very limited possibilities, and in a framework, socio-political framework and economical framework that is against us, completely against science. So, and because we also entered in a certain way, we entered in the wheel of the capitalism, making, you know, compete between us. For example, there was a paper, a physics paper in 1970s or something like that, that the authors discovered something, I don't remember, I don't have memory for these things, but discovered one very important thing that is extremely cited. And they say, oh, we can make alpha, beta, omega, and we put the names of the people because of the Greek letters, and then you will be the first, you will be the second, you will the ha, 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 ha. Now, who's the first author? Who's the second author? Who's the last author? Who paid the money? Who put the thing? Which kind of journal you are betting for? And this is journal of this. No, no, no. And now even the journals, they are creating spheres of power that are struggling about them, okay? And you see that sometimes, even in the moments in which you have to compete for a fixed place, they say, but you publish it in this journal, which is not good, or this kind of editorial, which is not fair. And you say, what? Sometimes you have this kind of fights that are just involved, and from my point of view, poisoning a little bit our own job and way to do the things. That's one point that is controversial. And then I don't know, I'm in some way I'm lost, because I'm making things, restoration, so on. On the one side, I would say, okay. On the other side, I would say, but give me the possibility to do that, please. More questions, more comments? No? Okay, we can, what time? Yes. Ten piedad.
Well, we've been like, I know science has to work on facts, data, and have some conclusions. But also, as you were saying, okay, we can also think about solutions, because that's also our responsibility, not just stay on facts and, our interpretations about those results, but also give these inceptions of further research or a little bit of hope sometimes. Sometimes when you read and understand and know about all the research, it's like sometimes for society, because it's a very key point, we need a little bit of hope from researchers. So how can you deal with that? Not being this, going into this alarmism that sometimes goes away from this fact and objective thing.
Here, that's a very also a very important point of view, which is when I have a subject, materia, in environmental science here. I give one that is basically biotechnology applied to the ocean. That's one of the, in environmental science. I don't know if I have here some student, they did it, okay? Is an optative one, okay? And then the first thing that I say to all my students is, we do have all the tools. We do have all the tools to manage this mess. From the socioeconomic point of view and from the, let us say, the political point of view. We do have it. We have, we can do it. However, I'm not a techno-optimist in the sense that I know that, sorry about that, but technology wouldn't save ourselves. It's a tool that we need to apply, but I don't think we have enough, we wouldn't have enough things to save ourselves from climate change, from the, from the resource deprivation, so from the collapse of the economy, from the collapse of, no, we do have to have in some way the maturity to say, this is not working. And then we have to change the model. We all have to change the model. But when you say that, if you say, I have to change the model, and then we have to make things in another way, automatically you are whatever. You are a communist, an anarchist, you are no, es verdad. I mean, it's truth. I mean, you're an outlier because the system is completely focused on one thing, concrete thing. So we do have for example, do we have the tools to make things different from the energetic point of view? Yes, but we have to lower the expense of energy. So that's the first thing. If we don't make that, it wouldn't be successful. And then you see that the people is building nuclear power, small nuclear power structures to feed the artificial intelligence. Okay. Or they take a lot of water, which is true, to just manage the situation of this artificial intelligence. And these things are counterintuitive for the maintenance of our own survivorship as humans. And then giving hope, yes, absolutely yes. This is what I say always. But then we have to think more locally and step by step, go by bottom up, to just try to figure out how we can make the optimization of these ways to respond. But it's not easy at all. It's not easy at all. More questions or comments?
In fact, it's a question from the audience that is online. I'm going to read. I think there is a lot of people out there who share the sensation for nature you have shared today. I do. But how do you think we can combat the economic interest posed by, for example, lobbies? Politicians often are just elected for 5 years and will not think of what you said generations after our grandchildren. Do you understand?
Perfectly. Okay. Look, why do you think China is betting for alternative energies? Why? Because it's fashion? Why China is betting for alternative energies and is shifting from carbon, gas, and petroleum to wind farms, solar energy, and so on. What is China? China is an autocracy, okay? It's like that, okay. And they have to look at 20, 25, 30 years. And in 30 years, we will be in a deep problem. So they understand that if they don't act now, in 20 years, more than probably, they will have to respond to their own people, saying, you knew that climate change was real, but you did nothing to stop it. Then I did something. The others make nothing. It's as simple as that. They have this long term. I don't say, and truly, I'm not saying, of course, no, the China's model is the perfect. No, it's not a democracy, come on. However, they have another completely different view of the world that we don't have. Especially, I'm telling Europeans, okay, Occidental people, let us say whatever you want. I'm talking about people in Spain, Denmark, or in the United States. And we do have these 4 years of action. And then in 4 years, look at Trump. Trump is trying in 2 years to make all the possible things to do not have to respond to their own electorate, to their own people in the next 2 years, okay, a pool that they have the next 2 years election. You know that is two, okay. It's 4 years the president, but then the chamber changes after 2 years. Is this the democratic way to go to the United States? So he has 2 years to make whatever he wants to do, which I don't know, I don't understand. Nobody understands, but it's a plan, of course. So that's important to understand. So the long-term view is very complex because we are all fighting each other to just make possible that we have our own singular benefit as continent, country, city, and individual. So we are comfortable with this point of, you know, at the end, we are all here, and we say, yes, the professor is right. Oh. But then we will make the same things that we make as usual because we feel comfortable. So how to readdress this? Not sure. I'm not a sociologist. There is one point which I think is really good, which is called the growth. And I know the growth from the Instituto de Ciencia y Tecnología Ambiental, in which I have been working for 10 years, more or less. And they have been working hard in the growth. And they demonstrated that the growth is a possibility. It's not perfect, no. But it's not destroying the economy or not destroying the human beings. It's just think about how to calm down the things that we are doing. But on the same time, we have one of my colleagues say, if one guy from the ancient Greece came here, he will think that you are gods. Because with your mobile, you ordered a motorbike and it comes to your house. And for the Greek guy of the 5th century before Christ, we say, you are God. How this is possible? It came a guy with your food. So we are gods. But being a god has their price. The price is basically the energy and the societal problems that we have of the structure of the society that we are witnessing. So it is possible from my point of view, yes, but we have to start thinking in another way. And we are start thinking in another way. And I think you, the next generation, start thinking in another way. And this starts to make desperate the true capitalist that is saying they are not consuming as we want. They don't want to have a car. They don't want, I say from my own sons, I have 2 daughters and 1 son. They don't want to have a car. They don't want to have a house. No, I will just try to make a house with my friends and with people, and I will try to share, and I will go with my bike. So I don't care about all this. And it's truth. And all the friends and their cousins are start thinking in another completely different way that we are not capable to do. My generation is very difficult because we lived in another completely different context. These things will evolve, and evolving, we will see what happens. But this will depend on us, but will depend on you, not on us. I mean, we can help, but it's the generation from the 20 to the 40 years, 35 years that has the, the stick of, in some way, responsibility to do that. More questions? Okay. Vale.
Pessoal, só falando aqui da lista de frequência, tá passando aqui, tá? Tem 2 listas. O pessoal que tá na disciplina da pós, vai estar seu nome vai constar seu nome aí. Se não constar, você acrescenta. E a outra lista é para quem não é da disciplina da pós, tá bom? Só para constar. Valeu.
Ahora, sí, ahora viene Carolina. A Carol, sí. A Carol, ¿te has presentado a Carol? Ah, no, vale. Ok.
Eu sei que não tinha enviado, mas eu vi.
¿Qué hora es? Ah, bien. Vale, vamos bien de tiempo. Sabemos. Muitos. Sí. Muito bem. Vale. I think some students asked to the director of the LABOMAR, Professor Lidriana, what is ballet? What is ballet? I say ballet all the time, sorry. Ballet is okay, but it's in Spanish. Well. Caroline Feitosa is a fishery engineer with a master and PhD in biological oceanography in the University of Ceará. She's a professor in LABOMAR at the Federal University of Ceará and coordinates the tropical ichthyology group and has expertise in fisheries resources with an emphasis on marine fish, particularly the following areas: population biology, ecology, and fisheries. And I think most of you know her very well.
That's with turtles, right? I will talk in Portuguese. I won't talk popcorn. No. Não. Bom, boa tarde. Como o professor Sérgio me apresentou, sou Caroline. Ele me passou essa incumbência aqui de falar da fauna associada às florestas marinhas, florestas de animais marinhos, né? Então, pela minha expertise, foquei peixes, né? Peixes, tartarugas, e no ecossistema recifal, que é o que eu trabalho, né? Desde sempre. Pode passar, Brau. Bom, suponho que vocês já viram bastante, né, classificação dos bosques, né, das florestas de animais marinhos. Aqui, só a título de ilustração, um dos nossos ecossistemas recifais, né, do Brasil, aqui é a formação recifal de Maragogi, alagoas, mas ao longo da nossa costa nós temos terraços marinhos, chapeirões, como em abrolhos, recifes bem distintos e com diferentes níveis de complexidade. Aqui as florestas de esponja, de gorgônias, os recifes de Cuba, né? Alguns trechos eles têm essas florestas de gorgônias, e os corais de profundidade, que são os corais que não formam recifes, né? Então, aqui são alguns exemplos de florestas de animais marinhos e que sustentam uma vasta diversidade de peixes. Bom, por que esses ecossistemas são importantes, né? Isso já foi bastante debatido. Disponibilidade de hábitat para uma infinidade de espécies, de vertebrados e invertebrados, favorecimento de biodiversidade. À medida que esses ecossistemas eles vão sendo degradados, essa biodiversidade vai sendo perdida. E tão importante, né, para a gente, em termos de suprimento alimentar, né, de segurança alimentar, fornecimento de recursos pesqueiros, e também para as outras espécies que estão associadas ao hábitat. Bom, então, o que a gente precisa conhecer sobre os peixes e tartarugas, né? Recentemente foi publicado no livro do professor Sergio Rossi o capítulo que eu acredito que boa parte dos palestrantes estão aqui representados, né? O professor Tito, Pedro, que falou mais cedo, Pedro Carneiro, Marcelo Soares. Então, eles fizeram um apanhado sobre o que se conhecia desses bosques no litoral brasileiro. É um termo antigo, né? Florestas marinhas tropicais, mas eu confesso que eu só ouvi falar com o professor Sergio Rossi, aqui no Brasil, né? Então, digamos assim, que no Brasil ele trouxe, né, ele inseriu esse termo para cá, embora a gente já trabalhasse com diversos ecossistemas classificados como tal. E particularmente importante esse tema agora, onde o Brasil vem pleiteando uma ampliação da sua zona econômica exclusiva, né? Então, já conseguiu no sul do país e agora na porção norte. E a gente vem debatendo a questão de exploração de petróleo na margem equatorial, discussão sobre inclusão dos bancos oceânicos em áreas, em unidades de conservação. Então, esse conhecimento ele deve ser gerado com uma certa urgência, para que a gente possa argumentar e tentar proteger esses ecossistemas. Bom, então, com essa publicação, né, tem-se o conhecimento, o estado da arte dos organismos construtores dessas florestas de animais, mas a fauna associada, o conhecimento ele é pontual, né? Então, fica já a dica aqui para futuras pesquisas tentando complementar essa publicação. Bom, então, entrando diretamente aqui nos peixes, né? Quanto mais complexo for essa floresta, maior será a diversidade de peixes. Isso é amplamente documentado na literatura. E não necessariamente por esponja ou assídia, o que importa mais nesse sentido é coral. Quanto maior a cobertura de coral pétreo, né, maior é a diversidade de peixes, né? Então, os peixes eles são ecossistemas, desculpa, são um componente super importante, né, de extrema relevância para as florestas marinhas tropicais, porque eles trazem nutrientes, eles controlam o crescimento de esponjas, de algas, para manter a integridade do ecossistema. Então, dentre os peixes, temos consumidores dos próprios invertebrados que compõem a floresta, formam a floresta, predadores e, digamos, arquitetos ou engenheiros do ecossistema, por falar pontualmente cada um desses. Então, eles acabam influenciando, por exemplo, na complexidade do hábitat, no funcionamento, à medida que eles vão controlando as populações das outras espécies. Aqui ilustrado é um pouco da biodiversidade da APA Costa dos Corais, a maior unidade de conservação marinha do Brasil. Então, aqui a gente tem alguns representantes, né, da rica fauna, que abrange uma área de 135 quilômetros de extensão. Bom, então, estou falando aqui já de peixes, né? Basicamente, os peixes recifais, independente deles estarem associados a um recife ou não, né? Associado a um substrato consolidado, já recebe o nome, passa uma parte do ciclo de vida ali, a gente já recebe o nome de peixe recifal. Então, tem espécies que são características do ecossistema, aqui como os snappers, né, os lutijanídeos, Liacioba, lame snapper, dog snapper, lutijano jocu, Algumas espécies endêmicas aqui estão ilustradas, como os escarídeos, que na verdade não na família Labrida atualmente. A espécie endêmica também de parrotfish, Labrida. Stegastes rocassensis, endêmica das ilhas oceânicas, né? E o peixe-borboleta. O peixe-borboleta é considerado espécie indicadora da saúde do ecossistema recifal, por se alimentar, isso é amplamente difundido na literatura, que ele se alimenta de pólipos, né, dos corais, mas ele também pode se alimentar de outros invertebrados. Mas é uma espécie indicadora da saúde do Recife. E predador também, né, considerado indicador, mesopredador, predador de topo, e também as espécies que controlam o crescimento de alga. Então, são espécies de peixes que, a partir da presença ou ausência, podemos inferir sobre a integridade do ecossistema. Bom, se conhece, né, de peixes recifais. Teve um trabalho que compilou essa riqueza, né, de Quimbaio e colaboradores, publicado em 2021, e tem no site do Ministério do Meio Ambiente. Então, são conhecidas 7.000 espécies no mundo de peixes recifais, sendo que 70% dessa riqueza está associada aqui à região da Indonésia e Austrália, na porção do Índico. E, no caso das regiões tropicais, né, do Atlântico e do Pacífico, são conhecidas 2.200 espécies, aproximadamente, e dessas, quase que 1.500 estão na porção do Atlântico, né? Então, a gente tem uma maior riqueza de espécies comparada ao Pacífico, aqui, que banha as Américas, né? E poucas espécies compartilhadas. Mas o interessante do Brasil é o alto percentual de endemismo, né? Esse endemismo favorecido pela barreira da foz do Rio Amazonas, que é considerado uma esteira, né, que conecta a diversidade entre Caribe e Brasil, Caribe sendo hotspot de biodiversidade, né? Então, é uma região de transição. Então, da fauna brasileira, exclusivamente brasileira, são conhecidas 408 espécies, e quase 30% dessas são espécies endêmicas, né? Aqui nessa imagem, as 4, né, são espécies consideradas endêmicas do Brasil, da província brasileira. Quando eu comecei a estudar peixe recifal, isso aqui ainda era reconhecido como espécie do Caribe, né? Todas essas eram as espécies do Caribe, estavam em processo ainda de descrição, que é bem recente, né? Tem uns 20 anos para cá. Bom, nesse ecossistema também é comum a ocorrência de espécies demersais, óbvio, né? Um ambiente bentônico, e algumas dessas espécies elas têm uma certa relação, uma dependência pela complexidade, né? No caso de garoupas, elas vão estar normalmente dentro de cavernas, enlocadas, então essa complexidade tridimensional ela é fundamental para o incremento da riqueza de espécies. Então, aqui, mesmo usando de camuflagem, né, eles têm no caso do linguado, ele tem uma associação com essa complexidade, né? Algumas espécies ainda assim usam de camuflagem, né, que não está tão clara, fica melhor, né? O peixe-trombeta aqui nas gorgônias se camufla aqui, e nos recifes menos complexos, né? Isso aqui, por exemplo, foi numa poça de maré, com as menores aberturas, né, reentrâncias, elas estão sendo colonizadas, né, aqui no caso da moreia-banana. E voltando para o endemismo, né, o exemplo das ilhas oceânicas do Brasil, principalmente das regiões mesofóticas, né? Eu costumo dizer para os meus alunos que cada mergulho no Recife mesofótico é uma espécie nova sendo descrita, né? Então, isso aqui foi de uma única campanha que pesquisadores brasileiros que estão fora do país coletaram, né? Na região de Fernando de Noronha, 4 espécies novas. Na verdade, isso daqui era um registro novo para a região. E espécie endêmica compartilhada entre as ilhas, né, Noronha, Atol das Rocas e o arquipélago de São Pedro e São Paulo. E aqui, no caso, né, não é um endemismo, mas é um padrão de colorido distinto, um morfotipo distinto na ilha do arquipélago de São Pedro e São Paulo, né, característico do isolamento daquela região. qual a importância, né, desses peixes? Os peixes dependem, né, dessas florestas marinhas tropicais e a saúde do ecossistema também depende da presença dos peixes. Algumas espécies, como no caso o surgeonfish, que no caso acanthurus quirúrgicos, é uma espécie herbívora que controla o crescimento algal. Então, é comum comentarmos sobre a mudança de estado no shifting baseline e mencionar que há uma migração, uma modificação dos recifes dominados por coral passam a ser dominados por alga. Essa não é a nossa realidade, porque nossos recifes naturalmente têm uma maior cobertura algálica, né? Mas, de certa forma, precisa ter espécies que controlem, caso contrário, não sobra espaço para assentamento de outros táxons, né? Então, aqui, predador, no caso mero, eles atuam controlando esses níveis tróficos. Também são fundamentais para a ciclagem de nutrientes. Espécies de grande porte que realizam migrações favorece essa troca de nutrientes entre diversos ecossistemas e também favorece a produtividade primária, né? Vai favorecer a base da cadeia alimentar. Bom, então, enquanto os engenheiros de hábitat, um exemplo clássico, né, de espécies que ocorrem no Caribe, essas aqui ilustradas, essa aqui é endêmica, né, do Brasil, que é o budião azul. Então, eles, além de atuar, né, nesse controle da alga, ele também raspa, né, a matriz calcária. Ele também raspa, né, acaba ingerindo o tecido vivo do coral. Isso é super comum em campo. A gente pode observar essas cicatrizes que ficam nos corais. E essas espécies elas têm uma válvula faringeal, elas trituram esses pedaços, esses fragmentos carbonáticos que são ingeridos, e elas são importantes na produção do sedimento carbonático. Então, é importante em dois sentidos, né? Então, tem dois serviços aí prestados, né? Tem os gobídeos também que se enterram e vão alterando aos poucos essa estrutura do sedimento. Do sedimento não, do ambiente. Vocês devem ter visto isso pela manhã, né, das interações com os ecossistemas, né? Então, essas espécies, elas utilizam, né, o hábitat dessas florestas marinhas tropicais, seja para se esconder de predador, seja como área de reprodução. Isso é muito comum nos recifes mesofóticos, na quebra da plataforma. As agregações reprodutivas, né, geralmente as espécies de maior porte, elas migram para a quebra da plataforma e isso é reconhecido como áreas de reprodução para algumas espécies, inclusive de importância comercial, né, que é um impacto da pesca considerável nessas espécies. As interações tróficas, né, onde muitos peixes eles se alimentam intensamente das espécies que formam o ambiente, né, e controlam tanto a distribuição como também controlam a abundância, né? Então, ambientes que são impactados, né, quando tem remoção de espécies que predam ouriço, né? Então, o ouriço se desenvolve rapidamente, aí vai impactar na cobertura algal. Então, tem toda uma relação que precisa de equilíbrio. E aqui as relações simbióticas, né? Essas relações elas são clássicas, né, nos recifes, né? Então, independente de profundidade. Então, todos os recifes eles apresentam, né? Durante um mergulho é fácil você observar essas interações, né? Aqui no caso, o Elacatinus, no caso Fígaro, né? Elacatinus fígaro, que nos recifes costeiros, não de ilhas, realizando limpeza aqui no Ampiraúna, no caso Maragogi. E essas três associações foram em Fernando de Noronha, né, que tem praticamente umas 15 piraúnas, né, que estão acompanhando o polvo enquanto ele se alimenta, para tirar proveito, né, de uma presa exposta que não foi ingerida. E nesses outros dois exemplos, né, diferentes mergulhos, a raia, né, enquanto se alimenta sendo perseguida, né? No caso, é uma espécie com comportamento seguidor que tira proveito também desse comportamento da alga, né, de forragear substrato. Aqui são duas espécies de cangulo preto, né, são muito comuns nas ilhas oceânicas brasileiras, E, no caso de Fernando de Noronha, tem interação com os golfinhos também, né? Então, o golfinho simula, né, o menor sinal de que vai vomitar, eles já estão ali prontos para comer. Então, eles comem tanto vômito quanto fezes dos golfinhos, né? Então, eles são bem oportunistas. As tartarugas, né? Para a gente ir para a parte que importa, né, dos impactos, são conhecidas 7 espécies de tartarugas. Dessas 7, 6 estão ameaçadas, em diferentes categorias de ameaça, e dessas, 5 ocorrem, né, no Brasil. No Brasil não ocorre essa clássica, que é aqui, que é da Austrália, né? E essa daqui que é dos Estados Unidos. Bom, essas espécies elas não são residentes, né? Mas passam uma parte do tempo, né, nos recifes e são importantes também para controle algal. Controle algal e controle de esponja, né? Mas também elas são importantes na transferência de nutrientes entre diversos ecossistemas, uma vez que, por exemplo, Dermochelis coriacea é uma espécie com hábitats mais oceânicos. Bom, mas quem realmente importa, né, são 2 tartarugas, no caso a tartaruga verde, que quando adulta ela tende à herbivoria, né, quando jovem é onívora. Tem preferência pelos prados de angiospermas marinhas, mas os prados eles estão conectados aos recifes, né? Então, consequentemente, ela fica entre um ecossistema e outro, onde nos recifes ela tende a se alimentar de algas, né? Então, controla o crescimento algal e indiretamente favorece a saúde dos bosques, né? de animais marinhos. E a tartaruga de pente, que controla o crescimento das esponjas, né? As esponjas, algumas delas são tóxicas, então não é todo organismo marinho que está apto a predar a esponja. Mas aqui nós temos 2 espécies que ocorrem, né, no Atlântico, o Olacanthus ciliaris e o Pumacanthus paru, que predam sobre as esponjas, né? Só que eles tendem a ser espongívoros quando a cobertura de esponja é maior. Então, no Caribe, ele é o clássico espongívoro, mas no Brasil, 70% da dieta é composta de alga. Mas pode se alimentar de esponja também. E as outras tartarugas, elas atuam como fertilizadores, né, conectores entre os diferentes ecossistemas, onde elas são espécies carnívoras, né? Então, elas se alimentam basicamente de invertebrados e de peixes também, mas sem tanta relevância quanto as 2 primeiras nos bosques de animais marinhos. Bom, agora um pouco dos impactos, o professor Sérgio já mencionou, mas só pontuar que muitos dos impactos que afetam os bosques indiretamente vão afetar os peixes e vice-versa. Isso é tanto, né, é claro já para todos. Então, o caso das mudanças climáticas, só achei interessante essa imagem, eu não vou discutir sobre isso, porque isso já foi amplamente falado, é que os eventos eles estão cada vez mais curtos e mais intensos, né? Então, eles estão... Antes eram escala de 10 anos, 8 anos, e hoje vocês podem ver um intervalo curtinho aí de 4 anos, né, entre os eventos, e observem que, com o passar do tempo, ele vem ficando mais severo. Então, essa última onda, aqui no caso do Brasil, né, essas ondas de calor que assolaram os nossos recifes costeiros, então, muitos, né, tenho falado bastante aqui de Maragogi, que eu trabalhei lá, mas cobertura coralina de Maragogi declinou consideravelmente, né? A maioria morreu. Então, já muda completamente o ambiente, né? Então, morreu uma colônia de coral, rapidamente ela é colonizada. Bom, práticas de pesca destrutivas, né? O professor Sérgio falou aqui do arrasto, né? O arrasto é um impacto invisível para a sociedade, né? Só quem trabalha na área sabe do impacto de uma pesca de arrasto. A gente tem cerca de 95% das nossas plataformas continentais já arrastadas, né? E é comum uma analogia com o desmatamento, né? Só que é completamente diferente, né? O desmatamento, desmatou, você tem que esperar crescer, né? No caso do arrasto, as regiões são arrastadas inúmeras vezes sem dar tempo da comunidade daquela região se recuperar, né? Então, a gente tem um relato dos bancos oceânicos, aqui em frente ao Ceará, de uma embarcação russa, se não me engano, que eles fizeram 80 arrastos em cima do topo de um banco oceânico. Então, eles destruíram tudo. Então, quando você fala em jardins de esponjas, jardins de gorgônias, né, eles não vão arrastar em Recife, né? Óbvio, né? Não tem como passar uma rede ali. Mas nesses outros ecossistemas, tranquilamente, né? Isso é feito e é desemaranhado, né? É desmontado aquele ecossistema. O que é importante, que é a tridimensionalidade, a complexidade, isso é perdido com essas práticas de pesca destrutivas. E aqui é uma arte de pesca, que é a arte de pesca mais pesada por metro quadrado, a diária, que são as dragas para capturar molusco, né? No caso, as vieiras. Então, elas, além de serem pesadas, que elas vão entrar bastante no substrato, elas têm esses dentes. Então, arranca tudo, né? Tudo que é de invertebrado. Então, pesca de vieira, ela é feita dessa forma. É uma modificação de arrasto. E ainda nessas redes de arrasto, eu devia ter colocado aqui, tem umas redes que a área de baixo ela é mais curta, e aqui tem correntes. Essas correntes elas penetram até 10 centímetros no substrato. E essas correntes elas facilitam a captura de flatfish, né? Linguados, né? Linguados. Falei o nome agora, uma pescaria importante que tem nos Estados Unidos, Sérgio, Canadá, Halibut. Então, bastante utilizada para capturar essas espécies que estão em íntima associação com o substrato, né? Então, a depender do tipo de floresta marinha, ela é 100% danificada. E associada à pesca, né, não podia deixar de mencionar, né, o efeito cascata na teia trófica. Esse é um fenômeno discutido por cientistas pesqueiros desde 95, 96, se eu não me engano. Ainda há pesquisador que questiona, né, mas esse impacto, se a gente monitorar aqui numa escala de 5 anos, aqui no Mucuripe, a gente vai ver, né, isso acontecendo, né? Então, hoje a gente já exporta herbívoro, né? Hoje a gente já come espécie herbívora. Então, não tem mais nossos recifes, né, carecem de predador, né? Dificilmente a gente encontra numa amostragem um predador, é algo bem raro, depende muito do local, né? E Jeremy Jackson, né, depois veio com essa síndrome, né, do shifting baseline. O que seria, né? Seria uma alteração de estado, né, do estado natural, do estado pristino, onde praticamente não tem nenhum ecossistema nessas condições. O shifting baseline, por exemplo, eu comecei a mergulhar em 97. Eu tinha uma visão daqui da risca do meio, né? Vamos supor que algum de vocês comece a trabalhar naquela região, o padrão de vocês já é completamente diferente, né? Então, às vezes, a gente trabalha com área controle e tratamento, e o nosso controle, na real, não é controle, né? Já é um estado completamente diferente do que era antigamente, né? E isso, quem trabalha em determinadas regiões há bastante tempo, isso é frequentemente visto, né? Se a gente for estudar nas nossas pescarias daqui do Nordeste, já houve mudança das espécies de vermelhos, de lutigenídeos, que são capturados, né? Então, guaiúba não era pescada, ariacó não era, lane snapper, yellowtail snapper, eles passaram a ser pescados, né, recentemente. Eles não eram o alvo, né, na década de 60, 70. Ainda degradação de hábitat por diversos fins, né? Poluição de diversas formas, plástico, que já vem sendo amplamente debatido, né? Os poluentes orgânicos persistentes também é um problema que precisa ser debatido, né? Enfatizado. Se fala muito em metais, né? Mas os poluentes orgânicos é tão problemático quanto. E sedimentação, né? Outro problema para esses ecossistemas. Eu não sei fora do país, mas no Brasil tá triste, tá difícil, né? Com PLs aí de desmatamento e supressão de mata ciliar, e tudo que poderia atuar contendo, por exemplo, poluentes da agricultura e da pecuária, né? Então, o destino a gente já sabe, né, qual é. E, por fim, as espécies exóticas, né? As espécies que, no caso ilustrado, o peixe-leão, que já é uma espécie considerada uma espécie invasora, que já também Fernando de Noronha, e já ao longo da costa nordeste, né, já amplamente disseminado, e para as florestas, né, propriamente dita, o coral-sol, que as ações, né, para a remoção dessa espécie invasora não têm surtido efeito, né, ela é bem problemática. No processo de remover, ela já vai ali se multiplicando, né, digamos assim, então, sem muito sucesso. Aqui eu acho interessante a reflexão, não é para polemizar sobre mudança de transição energética, né? Não necessariamente precisa, mas a gente precisa que seja justa, né? Mas não é o intuito não é debater isso aqui, mas só para mostrar aqui na nossa região, né, como esse litoral norte aqui ele vem sendo prospectado para a instalação de empreendimentos de eólicas offshore. E aqui tem um trabalho nosso, né, saiu daqui do LABOMAR, do Pedro, ideia do Pedro, que ele mostra a conectividade dos recifes do Caribe, aqui pela esteira de diversidade dos recifes do Amazonas, e aqui seriam os nossos recifes, né, os recifes da costa semiárida. Vocês podem observar, esse mapa ele foi gerado com informações de conhecimento nosso. Então, foram habitats de fundo consolidado mapeados, o que não quer dizer que não tenha mais, né? A gente pode ter ainda uma vasta cobertura desse substrato ao longo da nossa plataforma e que a gente ainda não conhece, o que é muito provável. E aí vocês podem ver a sobreposição desses empreendimentos com esses substratos, né? Com esse tipo de substrato consolidado, na maioria das vezes. E outra região também que causa muita preocupação é o Rio Grande do Norte, onde a gente tem formações recifais pouco estudadas e bem desenvolvidas, na região de Touros, né, do Rio do Fogo. E aqui o outro tema, né, a gente não vem muito aqui para o Ceará, porque está mostrado somente a ecorregião da região amazônica, né, que é para a vulnerabilidade de hábitats para a prospecção de petróleo, né, petróleo e gás. E boa parte, né, bem associada aqui ao Rio Amazonas, ao deságua do Rio Amazonas, ecossistemas bem sensíveis, né, embora a justificativa seja de que é bem, né, a prospecção, né, os estudos de pesquisa seriam em região bem distante da Foz, né? Então, isso é uma realidade, né? Então, a gente tem que trabalhar, conhecer essas áreas, conhecer o potencial, conhecer essa biodiversidade para trabalhar com cenários, né? Isso vem sendo feito, capitaneado pelo professor Alexandre Turra, se eu não me engano, da USP, né? Mas é uma realidade também. Eu comentei sobre os bancos oceânicos, eu acho que eu não tenho, talvez lá no início, mas também existe esse interesse, né, dos bancos que estão aqui a partir de Fernando de Noronha até o das Rocas, cadeia de Fernando de Noronha e a cadeia norte, né, de proteção, de inserir em unidades de conservação, para evitar justamente o arrasto nesses topos, né, que tem muito bancos de gorgônias, né? Então, para evitar o arrasto nessas regiões e também para banir qualquer possibilidade de prospecção de blocos de petróleo, né? Então, tem essa discussão. E aqui, né, o impacto se assemelha, né, se sobrepõe nos peixes recifais, só que no caso dos peixes recifais, a sobrepesca, a captura de indivíduos que causa sobrepesca de recrutamento, sobrepesca de crescimento. Esses dois tipos de sobrepesca eles ocorrem, e o que a gente observa é um declínio, né? Então, acho que bem perto aqui, ainda na década do oceano, talvez a gente já vai estar buscando outros alvos, né? Então, já existe demanda de prospectar estoques de espécies menos importantes de vermelhos, né? Como no caso da guaiúba, yellowtail snapper, e lutejano sinagres, o ariacó. Né? Então, são espécies que já vêm sendo prospectadas para aumentar a exploração sobre. A captura da fauna acompanhante, o bycatch. Bycatch ocorre em todo tipo de pescaria, seja pescaria de armadilha, seja pescaria de lagosta, espinhel, arrasto. Eles capturam espécies que não são alvo em elevadas proporções, inclusive de espécies em níveis tróficos superiores, como no caso mamíferos marinhos. Pesca fantasma, isso é muito comum, principalmente quando não se conhece. Por exemplo, se tem uma marambaia, se tem um recife artificial, ou se não conhece o fundo, né? Se joga uma rede, se põe um emalho ou faz um arrasto, geralmente eles ficam ali presos, ou então simplesmente quando perde, né? Isso é super comum no segundo semestre do ano, aqui nas nossas condições, por exemplo, que tem mais vento. Então, geralmente, essas artes de pesca, a pesca de lagosta, a pesca com armadilha, perde muita arte de pesca, né? E elas ficam pescando ali por bastante tempo. E, em termos de gestão, se trabalha de forma a confeccionar artes de pesca que sejam mais... que se degradem com mais facilidade, né? Então, eles preferem de madeira, porque caso se perca rapidinho, né, ela vai ser degradada. A perda de espécies chaves, né, para a alimentação dos peixes de maior porte. A gente observa essas modificações quando você visita um recife e você não vê mais espécies de níveis tróficos superiores. Geralmente você tem espécies que predam vertebrados móveis em abundância, como os hemolídeos. Poluição, né, todo tipo de poluição, de plástico, né, compostos, como eu já mencionei, mais adiante, e aqui o desaparecimento das estações de limpeza. Então, se a gente pensar que aquela coleta do peixinho ornamental na poça de maré pequenininho não vai causar nenhum tipo de impacto, né? Então, a depender da frequência e da quantidade, você está removendo espécies que são limpadoras obrigatórias, né? Então, elas são fundamentais para a saúde do ecossistema, né? Então, algumas delas complementam a dieta realizando essa atividade de limpeza. Para as tartarugas, né, também ocorre muita sobreposição desses impactos, né? Talvez o mais conhecido seja o bycatch. Hoje existem dispositivos, né, em artes de pesca. Artes de pesca foram modificadas justamente para evitar essa captura, né, de mamíferos marinhos, de tartarugas. Tem uma visibilidade na sociedade muito grande, né? Pesca fantasma, principalmente de redes, mesma coisa, poluição. Choques com embarcação, muitos encalhes ocorrem por causa de desorientação, quando elas se chocam com embarcação. Aqui no Ceará, com kitesurf também, né? Há relatos de choque dessas tartarugas e precisa urgente, né, de um zoneamento dessas atividades, né, digamos. Perda de espécies chaves também, áreas de descanso, principalmente, isso é relativamente novo, mas muitas espécies de tartarugas, elas se alimentam e descansam em recifes mesofóticos, né? Isso só foi descoberto há pouco tempo com pesquisas com telemetria, né? E elas também dependem da limpeza, né, dos limpadores nos recifes. Isso aqui é amplamente conhecido. Então, elas dependem também, como outras espécies de peixes recifais. E, diante de tudo isso, eu acho que a gente vai debater mais um pouco, né, ao término, né, do que falta, né, do que falta conhecer, quais são as lacunas, mais um pouco aqui das estratégias de conservação, que o Sérgio também falou um pouco, né? Então, é muito comum mencionar as áreas marinhas protegidas como uma salvação de tudo, né? E aqui no Brasil, pelo menos, o Sérgio comentou no Mediterrâneo também, elas são pouco efetivas, né? Então, a gente tem que realmente trabalhar nesse sentido da efetividade dessas unidades de conservação um olhar diferenciado para isso, né? Muitas vezes elas são estabelecidas e sem gestão, sem monitoramento nenhum. As pescarias, esse é um problema clássico, né? Assim, no Canadá você vê exemplos positivos, na Austrália, até mesmo nos Estados Unidos, mas no Brasil é uma tragédia a gestão pesqueira. E aí a gente ainda está caminhando naquele olhar defasado de trabalhar com uma única espécie, estabelecer defesa, o tamanho mínimo de captura. Isso tem se mostrado pouco efetivo. Basta olhar para a lagosta, né? Desde a década de 60 tem um defesa e o recurso não recuperou. E eles continuam insistindo nisso, né? Então, isso deve ser repensado. As pesquisas, né, cada vez mais com metodologias, com ROV, com BRUV, com DNA ambiental, que ampliem a possibilidade de incrementar esse conhecimento em termos de riqueza e, claro, realizar monitoramento, né? Então, é muito comum se estabelecer ferramentas de gestão, de manejo e não haver esse controle, né? Esse monitoramento ele é fundamental para avaliar o funcionamento. Educação ambiental, isso o professor Sérgio já, né, no finalzinho da palestra, comentou sobre isso, dessa mudança de olhar, né, que a gente já vê isso nas gerações mais novas, e incluir a sociedade nessa discussão, o engajamento, que isso também vem sendo amplamente difundido com ciência cidadã, né? A gente tem trabalhado com controle de encalhe, de lixo, de espécie invasora, sempre associada à sociedade, né? Trabalhando com ciência cidadã para esse sentido. Então, fico à disposição para perguntas, comentários.
Bravíssimo, muito bem, muito claro e sobre todo você seguro que também ha visto essas mudanças das últimas 2 décadas que você estava trabalhando na água. Há perguntas? Agora é o momento de perguntar à professora, porque normalmente é ela que pergunta a vocês. Então agora é o momento de dizer, vamos a poner a professora em dificuldades. Nenhuma pergunta? Bueno, eu sei que tenho 3. Não são facilinhas, são facilinhas. Espera um momento, que são fáceis. Bueno, a primeira não sei, porque quando você faz todo isso de intervenção dos peixes nos ciclos biogeoquímicos, eu tenho bastante claro a parte de ingesta, a parte de isótopos estáveis, a parte dos estômagos e toda uma série de metodologias. Mas quando você faz a ingesta, ou seja, a popó, como se calcula isso? Eu não tenho nem ideia.
Bom, a gente normalmente está pegando o estômago e pega o intestino, né? Então, no caso dos escarídeos, não resta dúvida, porque não dá para avaliar o estômago, porque o estômago é indistinto, né, com o trato digestório, e normalmente a gente faz uma estimativa considerando todo o trato digestório e vê o percentual de repleção.
Entendo. Pero no se faz, por exemplo, isso de esperar, porque é muito importante, por exemplo, a produção de areia por parte dos profis. Não há ninguém que faz uma fotografia quando então intentas calcular. Isso se faz ou é muito qualitativo?
Não, faz assim, uma estimativa grosseira com base na área da mordida. Aí se conta quantas mordidas por minuto, vai lá a média, aí tem um percentual.
Você pensa que aqui, no Ceará e no Nordeste, se poderia fazer algum tipo de aproximação de historical ecology com as pesquerias? Ou se é feito já? Porque eu não sei. Ou seja...
Sim, já tem, é
já
possível e a gente precisa fazer mais, mas já tem algumas comunidades que já estão...
Mas normalmente que é atum ou são peixes de arrecife de corais? Ou seja, quando você vai na historical ecology mira além de 50, 60, 70, 100 anos, quando mira essa informação, está mirando, por exemplo, as capturas, como fazem essa avaliação?
É justamente o oposto, não tem para o atum, tem para as pescarias, né, para os recursos pesqueiros recifais.
Vale, isso seria interessante. Eu penso que também no Arquivo de Ciências do Mar há uma quantidade de informação de fazer, pois isso, 60 anos, uma coisa por estilo, que seria interessante fazer uma comparativa de algumas espécies concretas, estaria bem. E a última pergunta que tenho é,
qual foi a sua sensação a primeira vez que você viu no vivo peixe-leão?
Ele é bonito. É lindo. Está muito bem, viu? Está muito bem adaptado.
Boa tarde a todos e todas. Professora Carol, quando você falou que está sendo ineficaz esse controle, né, de manejo da pesca, citou a lagosta, que não está tendo resultados, né, já depois de tantos anos. Existe alguma outra ou outras alternativas que estão sendo pensadas ou propostas para que isso realmente possa trazer resultados, né?
A pesca da lagosta é um caso assim que...
A lagosta foi citado, mas eu pensei,
então, mas continuando, que precisa ser estudado, né? Porque 2020, 2019 não se proibia a captura de lagosta ovada. E isso é uma ferramenta de manejo amplamente para crustáceo, né? Como já era feito com caranguejo. Não se permitiu, né? E recente. Então, você protege o estoque reprodutor no período, mas você não protege o animal. É assim, não dá para entender, né? E agora, mais recente, eles colocaram ainda limite de captura, ano passado. Então, você tem um período de pesca da lagosta, e dentro desse período, se atingir uma determinada produção de X toneladas, eu não vou lembrar agora, não importa se você tem mais de 2 meses pela frente. Não para a pesca, né? Então, e aí tudo isso fragiliza, né? Porque aí você vê esses escândalos de seguro-defeso, né, de uma prática que... O pescador não tem culpa, né? Mas de uma prática que só piora, né?
Obrigada.
Olá, obrigada pela fala, foi muito legal, assim, bem com o que eu trabalho, então eu adorei, né? Mas eu queria te perguntar, eu ainda não estou muito familiarizada com a pesca aqui na região, né? E eu queria saber o que tu me diz sobre esse conflito que existe, que tu citou a pesca de arrasto sendo feita por pessoas russas, por barcos russos. Eu queria saber se existe esse conflito entre a pesca de arrasto de outros países e mais industrial com os artesanais, os pescadores artesanais, porque no Rio Grande do Sul aconteceu isso e foi proibida a pesca de
Foi
arrasto.
bem gerido, né? Acho que com a Mônica Pérez, se eu não me engano.
Eu acho que
foi com ela. Na verdade, esse é um relato antigo, né? Em teoria, eles não podem, né? Eles não poderiam estar aqui realizando esse arrasto. E a pesca artesanal, nossas jangadas, eles vão pescar no talude. Mas do talude para o banco oceânico mais próximo é algo em torno de 80, 70 quilômetros, né? Então, lá eu acho que eles já não... De jangada, não. Né? De jangada eles não vão. Mas conflito de pesca artesanal com outras embarcações, não. Não tem esse tipo de conflito, não. Hoje o conflito maior é com eólica e eólica, né? Aqui no Ceará, aqui na nossa região, é eólica.
É que lá no Rio Grande do Sul acho que funcionou muito justamente o proteger a pesca artesanal, por isso proibia, né? E então aqui nesse caso não daria certo essa ideia aí mesmo. Obrigada.
Muito bem. Bueno, hemos acabado este segundo día de la Spring School, la parte de seminarios. Ahora tenemos un coffee break para todos los que quieran tomar un cafetinho o un suco, una cosa así. Y después haremos una pequeña mesa redonda como ayer, hablando de algunos temas y algunas cosas. Intentaré no torturar demasiado a los palestrantes. ¿Ok? En media hora más o menos, a las 4, lembrando a gente que está na outra parte de online que não será traduzida a parte de a mesa redonda. Não será traduzida para eles, ok? Après the coffee break, sim. vamos a alargar más demais eso, porque aí falamos de algunos temas muito, digamos que, muito interesantes e muito centrados en problemas dos oceanos, etcétera, etcétera. A primeira cosa que eu queria, en realidad, fazer uma reflexão con vocês desde o punto de vista, temos alguna serie de, digamos que, de problemáticas ambientais concretas. Y esas problemáticas ambientales necesitan de soluciones, pero según a su opinión, ¿cuáles pueden falar tranquilamente en inglés o en español devagarinho, poco a poco? ¿Vale? Si ustedes falan en español poco a poco, se entiende también. ¿Cómo ustedes prefieren? ¿Vale? Entonces, la pregunta que tengo para ustedes es ¿cuál serían las prioridades según ustedes? O sea, imaginen que ustedes tienen realmente, que ya sé que es muy extraordinariamente utópico, pero que ustedes tienen la posibilidad de influenciar un poco o que sería las políticas, digamos, para los océanos, porque ustedes saben de que se ha feito esta década dos océanos, 2030, onde se están haciendo cosas efectivamente, mas aparte que aí falamos un pouco de iso, porque estábamos falando un pouco de iso, de esa, bueno, sí, hai coisas que se están facendo, mas, por exemplo, as COPs, pues parece que cada vez son máis remotas as soluciones globais, parece que pasan máis por soluciones multilaterales, mas non globais, etcétera, etcétera. ¿Cuál pensan que desde un punto de vista de ciencia sería o máis urgente, o que se vocês tivessem a posibilidade de fazer algún tipo de aproximação, algún tipo de digamos, a nivel más global, ¿cuáles serían as funções, cuáles serían os mecanismos que vocês intentarían activar en ese sentido?
I think that if I could have the possibility to really influence some politics or some politicians, I will try to give the message that we need to keep on studying and to obtain data, because for most of the ecosystems we still don't have data, and I think that we cannot protect anything that we don't know. So I could try to, if I could, I would like to try for more funding to keep on investigating this environment that we still don't know anything about. And then once we have the data, take a list of priorities or the most vulnerable ecosystems and start to try to protect them. It's something, I don't know, it's a topic, but I think that I will start to do this. Sí.
Tá. Bom, é complexo, né? O tema é difícil, como você falou, é beira a utopia, né? Mas eu vejo que, de uma forma geral, tem a boa vontade, claro, mas às vezes falta vontade política, né? E eu acho que o caminho de transição energética ele precisa ser amplamente debatido e dar alternativas, né? Eu não vejo alternativa. E mais recentemente o que a gente tem visto foi, por exemplo, a gente fala tanto do plástico, né, da poluição por resíduo plástico, e o nosso presidente não assinou, né, contra o uso do plástico. Só que assim, isso do plástico já está amplamente difundido. Eu acho que a gente precisa investir em ciência, em pesquisa, para a gente buscar estratégias. Porque como é que a gente vive sem plástico? Não é? Eu acho que é uma realidade, a gente sabe que é problema, mas a gente não tem como desistir do plástico ainda, né? Então eu vejo muito que são problemas que são reconhecidos, mas que a gente ainda não tem a solução, né? Difícil de ser implementado.
for me is it's more or less clear that the education and the beginning and the end of the situation, because the political are what is the interest of the decision maker or the politician are the vote. So we need to have a population that are educated, well informed about the situation of the ecological situation or what will be dangerous and so on. So I think education at the beginning, education at the end. And another way would be talk with the businessman and try to sensibilize these people that every action of the human make a print in the environment. So try to minimize or reduce this print for the business everywhere what they will do, or try to recover after that. I think these two actions I will be trying.
In line with that, en línea con eso, ¿vale? Que es muy interesante, tanto esta mañana como también un poquinho esta tarde, estamos hablando de o rol del cientista. Yo quiero que falemos un poquito más de eso, porque yo he falado, mas penso que también es, o sea, hace 25 años aproximadamente yo tuve una discusión con un profesor senior, yo era un doctorando, mas tuve una discusión porque ese profesor que trabajaba en esa línea de pesquisa, de investigation, de research, de pesquisa, entonces él decía que los cientistas no tenemos que hacer nada, absolutamente nada para comunicar con la sociedad directamente. Nosotros ya tenemos los papers. Ya tenemos los papers, nosotros estamos haciendo nuestro trabajo y que tenemos que mantenernos en esa ivory tower, en esa torre de marfil. Y realmente él estaba convencido de eso. Era una persona también, no es por desmerecer, pero era una persona mayor y tenía una mentalidad muy concreta de decir, no, yo eso no estoy de acuerdo. Yo en aquel momento estaba comenzando a hacer divulgación científica, que yo gustaba de escribir articulitos y cosas así, de ser periodista científico. Entonces, gustaría un poco de profundizar sobre eso. O sea, ¿dónde está, según ustedes, con tranquilidad, dónde está el límite? ¿O qué tenemos que hacer? ¿Cuáles son los mecanismos para realmente llegar a la sociedad en un momento que es muy complejo? Ya era complexo antes, mas ahora es mucho más complejo. No sé, ¿cuál es la seva opinión de eso?
Espero haber entendido bien la... In my opinion, I think that educating new generation is fundamental. I mean, I know that it's just like a first step, and it is not going to solve all the problem of the world. But I think that if you grow a generation of people that know about the problem of the sea, they know about the problem of marine animal forest in general, the loss of biodiversity, etc. You can also grow a generation of people that respect the sea and respect the biodiversity. I think that, in my opinion, it takes time, it takes energy and effort to do citizen science activity, education activity, but I think it's worth to do it. And it's not so complicated in the end to pass this message to the new generation. You just have to understand how to communicate with different ages, let's say, no? You have to have the correct language for children, the correct language for graduate, the correct language for every age, let's say. But I think it's really, really important to do it. And since it's not so complicated, it is worth to do this effort, in my opinion. And I have the direct feedback because I mean, I teach some courses at the university, okay, but I also did some, I talked about it a little bit this morning, but I also did some education activity with the students of like from 9 years old up to 15 years old. And they give me feedback that they are really interested. They knew things that they didn't know anything about, and just with 2 hours spent together walking in the sea, in the beach, and recognizing organisms. So it's not so complicated, and for me, it's really important to do it.
Eu concordo contigo, Sérgio, da necessidade, né? Eu acho que isso é papel do pesquisador, envolver, né, a sociedade. A gente faz muita pesquisa e não, a gente termina um trabalho, a gente fala de políticas públicas, a gente faz aquela consideração final bem bonita, né? Mas como transformar isso, né, para alcançar no gestor, a sociedade no geral, né? Então, eu acho que falta, de certa forma, esse envolvimento do cientista puro, né? Então, eu acho que submeter um projeto, hoje em dia, as agências de fomento, elas deveriam pontuar um tópico como esse, de tradução de ciência cidadã, de traduzir aquele resultado da pesquisa, né? De dar uma devolutiva para a sociedade, porque hoje é tão comum, né, se trabalha com comunidades pesqueiras e nem sequer retorna lá, depois que você coleta os dados, né? Então, ciência cidadã, hoje eu acho que é um caminho, mas eu acho que hoje, assim, eu suponho que a gente que tenha um engajamento, que vai haver um engajamento maior nos próximos anos e, somada a isso, uma mudança de olhar, né, da sociedade, né? As pessoas mais novas, elas já vêm com uma cabeça bem diferente, né? E eu acho que a gente poderia, eu vejo uma coisa boa nas redes sociais, eu vejo essa divulgação da ciência, né? Você vê os laboratórios aqui das universidades, as ONGs, então, assim, mesmo que seja pontual, seja ali a conta-gotas, mas chega um pouco de informação.
Yeah, okay. Yes, I keep this idea of the Marfield Tower. I think we are most of us are teaching, working in a educational institution. I think we need to be most agent of rationality when we affront the problem with the student. We need to be agent that polarize the society. You are good, you are bad, or something like that. So we need to understand that every country need to develop, every country need make business to give employment, to give in the deeper part to have a better life or better quality of life for the citizen. But this no need to be that everything is destroyed. We need to be agent to say, well, we need to have clear rules. This is when we need to show to the student that there is a border where you no need to cross because they began to destroy everything and it cannot be recovered. Or make in a rationality point of view that some part need to be also, or all the society need a space in to done the function, like the animal forests have a function. We need to be in this point of in this position for the student. I mean, more that we personally we can have a position in front of the reality, but For the student, we need to teach this rationality. If we are, in the contrary, agent of the polarization, we began to be part of the problem and not the solution. So I think need to be this, how we say, amalgama, for the situation in the environmental in every country. Some countries have more problem or other less. But I think this is my point of view.
Exacto.
So thank you. I partially agree with Americo, and I don't think the scientists should do too much divulgation. Let's say that it's important, the divulgation is done. So why you should put in the hand of someone that is not a professional on that? You don't ask to a private company, to an engineer leading a company of construction, you don't ask to do advertisement for the company. There's a section of the company that does advertisement. So if we think that it's important, we put professional in doing it. Because personally studied statistics, ecology, chemistry, mathematics, not communication, etc. So I can have, someone of us can have a personal gift on being able to do, but it's random. It's not good. Look at the documentary that I did. It is done by a professional. It's very good. It goes to exposition in international exposition. If I do myself with my GoPro, with something like that and put some writing, good, I spend time. So we are scientists. We are supposed to spend our time in solving big problems, in having idea that goes beyond the normal question. So we should not have also the task to communicate, to invent a way to speak to the kids, etc. We have to be available for the professional to do this. For example, in my opinion, if everyone says that it's important, the communication, okay, in every department, for every 10 researchers, there have to be a professional of communication that goes to the researcher, ask what are they doing, translate in the correct language, is trained for that. It's at the cutting edge of the media, social communication skills, and do something good. If not, you ask to do things that people that are not professional on that, and it is not, it means that it's not important. Above all, I mean, if we become, if sometimes we are asked to do this, we spend a lot of time doing communication and less science, but we receive a salary that is for solving the problem of society with applying an objective and standard and scientific protocol and method, and come up with solution and possibly other questions, and then someone else will do it. If we apply the division of tasks, everything works well. As I told you yesterday, I'm not going to transplant coral. I don't have to transplant coral. I don't have a PhD, 3 postdoc, and the salary of scientists to plant coral. I find a protocol that is viable, and then someone else goes to do this. The people that do volunteers for transplanting coral, they don't have to do an experimental design. No, it's not their task. They have to apply something. If divide the task, everything comes better. If we do everything, nothing's coming good. So I think that is not our task. There have to be professional for that, and that's it.
Okay, I'm a little bit in disagree with you, because you're putting the example of the movie. Who is the face that is explaining the movie?
I don't have to do the movie. I have to be available for doing something, but it doesn't have to come to me, because that is a product that it's cinema level product. I can never do that. So I can be available. I say, we have to be available to do things, but I cannot stay there thinking what is the best language to use with the kids. There is someone that said to me, so you have to be like that and explain me like it was I remember that when interviewed, they say, okay, explain me this as you were explaining to a 3-year-old kids. And then after said, now explain me for your colleague. So you can be available for this, but you cannot be the one that manage everything.
Yeah, I agree. I agree in that sense, but I think we are now like having problems of not just one solution, but you have like multidimensional scenarios now. So we have to find also multi-scenario responses and solutions. And I think what we are missing now is more collaboration and cooperation between these scientists and communication specialists and educators to have this multi-scenario and multidimensional response. It's not just one solution. It's not just about communication. It's not just science. It's not just marketing. It's not just politics. I think all these sectors, and I hope tomorrow I will talk a little bit more about this, but they have to communicate and they have to interact between. I know there is not just one sector responsibility, but we need more interaction between all these like multidimensional society that we have.
But it's the division of task. I mean, I have to give the scientific content because I'm trained to do this. But then I don't have to translate for people. It have to be someone good in this to translate on people. And this person does not have to take the political decision. You have to report someone to the politics, and politics have to take the decision. It's not me taking the decision, not him doing science, it's not the political doing the movie. But if everyone does its own things, this is the multidisciplinarity, working together. Of course, yes. If you don't communicate...
To answer to Lorenzo, I mean, I completely agree with you that if we can separate the task, everyone will benefit from this because we can focus on science and another person can focus on communication. But in some cases, you don't have this separation task simply because you cannot contract anyone to make your data more public also for non-scientific people. So not all the projects allow, with the funding that you get, to contract communication staff to do exactly the nice documentary that you show us. So in some cases, you have to do it by yourself. Either it will not be perfect, it will not be the perfect communication, maybe, but you do your best with the resources that you have. I think that in some occasion, it's better to do it than lose the possibility to communicate a good result to also non-scientists, in my opinion.
Yes, but it has to be an emergency. I mean, you say, if there's no other way, I can try to do something. It's like in your lab, you don't do the maintenance of the electric stuff. But if there's no one to do it, of course, you do it and you do what you do. But the things has to be a lab, there have to be someone that is a technician that is able to fix everything. If not, we can do everything, but we decrease the level. So yes, it's true that if there's not another way to do, we should do it, but we should tend not to do it because we should tend to a high-level solution. Because no, I can try to fix that, but I don't do a professional work. But of course, if there's no light and no one does it, I'm gonna do it. But it has not to be the normal case.
Ou seja, esta é a segunda pergunta real. Qual é a sua visão da sensação de urgência real? Que eu penso que estamos falando muitas vezes, como ecólogos que somos, de esos phase shifts, que são muitas vezes de golpe. O sea, você passa de um estado a outro de uma maneira brusca, de uma maneira completa, porque o sistema não aguanta mais e vai a um outro tipo de sistema. Então, nós como cientistas, em que posição estamos nesse momento para respecto a esse phase shift? Ou seja, sim, eu concordo bastante com o que está dizendo Lorenzo, que tem que ter umas tasks, mas ao mesmo tempo, alguma coisa estamos fazendo mal, realmente mal, para não conseguir transmitir a urgência do momento, real. Não uma urgência histérica, nem uma urgência não instruída, mas com todos os elementos que temos, uma urgência de dizer, isso é um problema, o que seja. Não é necessariamente só a mudança climática. Então, como de urgente veis vocês esse tema real de fazer o salto e, sobretudo, quando foi o momento
onde vocês se deram conta que o que estamos fazendo para comunicar com a gente, com as pessoas, incluídos os estudantes, quando vocês pensam que não, digamos que se tinha que fazer de uma maneira diferente, ou não se tinha que fazer de uma maneira diferente. Igual sou eu que estou errado, não sei.
Eu acho, Sérgio, que a maioria das vezes não é feito. Não chega nem a ser feito. Então, fica muito no âmbito da ciência, fica muito restrito, fica muito dentro de uma bolha. E, na maioria das vezes, assim, na minha área, assim, com o que eu trabalho, eu vejo que muitas vezes não é feito. É você chegar numa colônia de pesca e ele não saber qual espécie não pode ser capturada. Porque ninguém faz esse papel, né? O órgão fiscalizador diz que não é atribuição dele, o Ministério diz que não é dele, não é atribuição da colônia, e é de quem? Você espera que o pescador vá no Diário Oficial da União? Né? Então, assim, isso é o mínimo, né? Você chegar no desembarque pesqueiro desse e conversar com essas pessoas, né? Explicar por que aquela espécie... pescou, tudo bem, não tem como evitar que seja capturada, mas explicar o porquê, né? É o mínimo de conhecimento, de transmissão de conhecimento que não é feito, né? Eu vejo muito disso.
Boa tarde, pessoal. Meu nome é Enio Sombra, eu sou professor há mais de 20 anos e eu vejo essa problemática, né, da divulgação científica já há muito tempo, né? Então, assim, hoje nós temos vários divulgadores científicos, então realmente já está melhorando, né, entre aspas, mas a gente precisa ainda fiscalizar bastante, né, porque tem muita informação, mas ter muita informação não quer dizer que ela é de qualidade. Aqui no Brasil, a gente tem a educação ambiental ela sendo vinculada a outras disciplinas, né? Aqui ela tem que ser vista, isso de acordo com a lei, como um tema transversal, né? Então, assim, eu particularmente já conversei com alguns políticos tentando a gente levar um projeto para tentar desvincular isso e tornar essa disciplina, né, uma disciplina independente, porque atualmente a gente precisa de um protagonismo maior, né, na educação ambiental, por tudo que a gente tem passado, né? Então, assim, a minha concepção é que toda a problemática que a gente tem hoje, ela é de cunho de ações humanas, né? Ela é resultado de ações humanas. Então, né, parafraseando o Paulo Freire, a gente tem que a educação não muda o mundo, mas ela muda as pessoas e as pessoas é que mudam o mundo. Então, vamos atuar, se a gente quer algum progresso, né? Isso é a minha concepção, mais uma vez falando. Se a gente quer algum progresso na crise climática, eu acho que a gente tem que atuar nas pessoas, né? Na conscientização das pessoas. E eu senti muita resistência, né, dos políticos aqui em tentar implementar esse projeto de tornar a educação ambiental independente, né? Para ela ter um protagonismo maior. Não, tem que ser federalizado, isso é lá em Brasília e tal, então realmente não foi para frente. Eu gostaria de saber de vocês, né, dos pesquisadores que estão aqui, que não são, inclusive o senhor, professor, que não são aqui do Brasil, né, como é que é a educação ambiental no país de vocês, nos países de vocês, como é que vocês tratam essa temática, né, e a questão também da divulgação científica, né, como é que está por lá, né, a gente volta aí lá para o Carl Sagan da vida, que teve um programa que influenciou Neil deGrasse Tyson, que é um dos grandes cientistas hoje em dia, ele abertamente fala que foi influenciado pelo Carl Sagan. Então, assim, eu queria um pouquinho da experiência de vocês, nos países de vocês, como funciona a educação ambiental lá e também a divulgação científica. Obrigado.
Él hizo una pregunta directamente a vosotros, muy simple. ¿Cómo veis vosotros en vuestros países fuera de Brasil el tema de la educación ambiental? ¿Cómo está progresando? ¿Cómo está yendo? ¿Cómo creéis que está evolucionando en general?
Si por mi país, entiendo Francia, que es donde vivo ahora y hago este trabajo, creo que hay mucha iniciativa privada para asociaciones que se dedican a la divulgación. Y, por ejemplo, un ejemplo que yo trabajo con Ander de Paul, hacemos una expedición científica donde yo llevo el topic ellos me han dicho, nosotros tenemos que hacer divulgación, etcétera, dame un topic. Hemos puesto en la expedición mi topic, yo meto la ciencia y ellos hacen, estoy a disposición, estoy en la entrevista, obviamente voy a hablar con pero ellos organizan todo y salen con mucha divulgación, algunos vídeos, algunos documentarios, pero sobre todo llegan a las Naciones Unidas con menciones porque tienen un despacho con gente que se ocupa de la divulgación para la escuela, se ocupa de la búsqueda de fondos, por ejemplo, del plaidoyer, que es de la presión y de llevar una idea y de la lobbying a la UNESCO, a las Naciones Unidas, etc. De esta manera tienen una división de trabajo y ellos trabajan, como decíamos, ¿no? Trabajan con científicos porque no hacen una expedición donde hacen vídeos y ya está, de cosas así. Meten una verdadera expedición científica de la cual ellos no se ocupan. Yo tengo mano libre sul topic científico, ellos me meten a disposición, tienen técnicos, buceadores, etcétera, pero sobre todo hay 3, 4 personas que se ocupan de venir allí. Y cada vez en el barco, en la expedición, hay un artista, que puede ser un artista, un pintor, etcétera, que tiene una idea y viene allí. Un influencer, como hemos hablado de esta Camila Etienne, de otro, o personas una escritora que ha escrito un libro, etcétera. Y así yo pienso que yo hago una cosa, ellos hacen la otra y todo viene de mayor calidad. Ellos no se meten en la investigación, que no saben de qué trata. Y yo no me meto en el lobbying, en la plaidoyer, no sé cómo se traduce el plaidoyer, llevar una moción. Y todo funciona bastante bien. Y hablo de la experiencia personal, pero veo que hay muchas asociaciones privadas que hacen esto. Y ahora, por ejemplo, también el CNRS, con retraso, el público llega siempre un poco retraso, el CNRS empieza a ver cuáles son sus científicos un poquito más divulgables y vienen allí y te piden si puede hacer una entrevista, se puede darle las fotos, pero yo les doy las fotos, son ellos que luego deciden cómo utilizarla. Yo me meto allí y ellos hacen un video, un documental de 8 minutos con el mismo formato que describe. Y está empezando también el público con mucho retraso, como siempre es estas cosas que el público tiene un poco de retraso. Pero el tema del hecho que, bueno, que no, como hay, es triste decirlo, pero como hay plazas fijas, yo doy una posición a alguien hace 10 años, se queda 20 años, se queda atrás con esta tecnología, con esta forma de comunicación. Entonces, si hay una persona que es cerca de retirarse para la comunicación, todavía utiliza Facebook y ya está, o la prensa. Y no, entonces, mientras una compañía privada es mucho más ágil en esto y más al día. Pero funciona, me parece, bastante bien.
A ver, ¿en inglés o en español? ¿Es igual? Español, vale. Ahora, a nivel de educación ambiental y sobre temas oceánicos, el último programa de la Comisión Europea venía con 3 proyectos enfocados a escuelas azules, con el apoyo también de la UNESCO. Entonces, está dentro de Mission Ocean, que es esta gran misión de la Comisión. Había una parte destinada a Blue Schools. Entonces se ha hecho la red europea, bueno, está la red europea para tú aplicar a hacer una escuela azul, con un apoyo hay 3 figuras de financiamiento para que las escuelas que tenían un interés en integrar temas oceánicos a nivel europeo lo pudieran hacer. Entonces daba una ayuda de 10.000 euros, era al máximo, y esto ha hecho un repunte muy alto a nivel de escuelas azules a nivel europeo para incluir la temática de Ocean Literacy. Y empezando eso, por interés de las escuelas, desde de integrarlo dentro de su currículum, aunque todavía no hay algo como aquí en Brasil, que ha sido un decreto y que está ya instaurado por ley, pero sí que, por ejemplo, Portugal ha sido pionero en toda la integración de la cultura oceánica dentro de las escuelas y la educación ambiental, pero se ha hecho ya a un avance porque durante mucho tiempo la educación ambiental había sido muy a nivel no formal. Y ahora se está intentando hacer ese paso a la educación formal. Y hay escuelas, nosotros desde la entidad donde yo trabajo participamos con una Blue School y puedo decir que el trabajo que se hizo que son institutos que están acostumbrados a trabajar sin dinero. Tú das 10.000 euros y hacen magia. Y realmente ves que hay un impacto muy grande en toda la estructura de la institución. Entonces yo creo que es un impulso, que hay cosas que quedarán, otras actividades que no se pueden hacer, pero esta parte de la educación ambiental, creo que con este impulso que viene desde arriba, con una policy desde la Comisión Europea por apoyar la educación en la cultura oceánica, pero que beneficia al bottom-up, ¿vale? Que beneficia al bottom. Y esto ha hecho que realmente ahora haya un empoderamiento muy grande para promover la cultura oceánica y la educación ambiental en Europa.
Però penso, perché ora mi viene alla mente también esperienza negativa, que todo está, tiene el problema contrario también, que a veces parece ahora que es más importante comunicar lo que hace más que lo de que hacerlo. Yo he tenido experiencia negativa en esto con un proyecto que llevaba sul refugio profondo de las Gorgonias, que necesitaba varias partes con financiamientos per genética, etcétera. Ha cogido la leadership, un fotógrafo, muy buen comunicador, que ha sido, yo no sabía casi nada, él ha pedido dinero a la Agencia de la Biodiversidad francesa, súper contenta de darle el dinero. Hemos empezado el muestreo, él ha hecho un trailer, os puedo mostrar, el trailer es bellísimo, con algunos muestreos, los primeros 3 días de muestreo, y luego todo el dinero se ha utilizado por esto, y yo no he sabido más nada. La Agencia de la Biodiversidad un día me dice, ma este proyecto, y digo, yo no estaba en el proyecto tampoco. Tenéis un buenísimo vídeo, un teaser, trailer, fotos a UNESCO, pero el trabajo nunca ha sido hecho. Y digo, yo no puedo hacer el trabajo, yo necesito 30.000 euros para la análisis genética de paternidad, la conectividad. Claro, cuando has hecho todas las fotos y todos los vídeos, ¿por qué estar allí otros 3 años a pagar la genética? Ya está comunicado, ya está dicho que es un grande proyecto que salvará las Gorgonias, etcétera. Ya no importa hacerlo. Y esto está pasando mucho. Sí, 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 sí. Sí, sí. No.
Solo aclarar que hay que hacer la diferencia entre comunicación y educación, porque lo que tú hablas es comunicación, pero se tiene que diferenciar de educación. Y eso, a nivel europeo, lo que yo mencionaba era esto de las escuelas azules, y estoy totalmente de acuerdo que para hacer una buena comunicación tiene que haber una base científica y tiene que haber un trabajo científico detrás.
Ahí, sí. Déjala acá. Ya. No, bueno, ahí respondiendo a las dos preguntas, un poco con lo que hacía Sergio y del colega de allá. Bueno, yo también me he hecho mucho tiempo esa pregunta, Sergio, ¿qué estamos haciendo mal? Pero de ahí hablando con periodistas, el problema es que nosotros no entendemos lo que es la comunicación. La comunicación se actúa de 2 partes. Entonces, por más que nosotros estemos hablando, si la otra parte no tiene el interés de escuchar, no lo va a escuchar, ¿ya? Aunque nosotros hablemos, hagamos formas para poder explicarlo, no va a ocurrir, ¿ya? Y digo un poco enganchándome con eso, en caso de Chile, hay un programa en el cual yo trabajé que justamente trabajaba con la educación ambiental, o más bien desarrollo de competencias científicas, ¿ya? Y dentro de eso, obviamente, entra la parte ambiental que es Explora, ¿ya? Ya se ha permanecido por mucho tiempo, ¿ya? Pero el problema va de nuevo en el interlocutor, ¿ya? En algún momento viene el nivel de las prioridades, ¿ya? Entonces, por más que mucha gente sale con una buena formación en el tema ambiental, cuando llegas al minuto de decidir, pucha, tengo que vender este producto que yo sé que está en veda, pero tengo que comer, no hay educación ambiental que resista el punto. Y yo creo que y eso hace como diferencias entre los distintos países europeos. Ya están en la pirámide de necesidades, ya cumplieron, no tienen necesidades de vestuario, cumplen sus necesidades de comunicación. Entonces, ellos están al nivel de poder priorizar la parte ambiental. En el caso de Chile, muchos de mis estudiantes tenían que decidir o cómo, o vendiendo centolla ilegal, o no como. Entonces, ese es el gran dilema, por lo menos en Chile, siendo que el gobierno, el Estado, invierte permanentemente y constantemente en hacer una buena educación ambiental. Ellos lo saben, pero las prioridades son más fuertes a veces y eso es lo triste. Así que eso.
Entao, ¿por qué, dentro de todo este panorama, por qué es tan difícil? Sabemos todos que los océanos son a base de toda vida en la Tierra. ¿Por qué es tan difícil falar de océano y por qué es tan difícil que a gente comprenda la importancia del océano? ¿Por qué?
Io penso che è perché non lo stanno haciendo unos profesionales, perché la mayoría de la gente que hace la divulgación, que habla de esto, son científicos que no saben cómo se hace. Puede ser más bueno, menos bueno, pero si tú metes un director de cine a hacer un spot su una cosa, viene bien. Y si no, significa que no es bastante importante, entonces no lo haces. Seguro. Y si metes ya verás que se mete unos publicitarios a hacer una divulgación, una comunicación, sale el tema. Pero no lo están haciendo unos profesionales, no da dinero. Si tú ves un proyecto, hay una pequeña parte que luego la utiliza un poquito más sobre la divulgación.
Pero lo ves muy lejano el océano. ¿Por qué lo vemos tan lejano? ¿Por qué? No me digas, no. También en nuestra ciencia.
No.
De 30 proyectos, 1 es de océano y 29 son terrestres. ¿Por qué?
Bueno, significa que la prioridad es su la Tierra. Esto pero lo que dan los proyectos, otra vez, son los políticos, son la lobby, son la presión. Puede ser que los biólogos terrestres son mejores en dar importancia o hay más interés económico.
Eu acho que é porque não é visto, né? Não é visto, é difícil de visualizar, é difícil. Você vê um desmatamento, você vê, né? As árvores sendo cortadas, um arrasto de fundo você não vê impacto. Uma espécie está diminuindo no oceano, se não tiver alguém estudando, você não vai perceber. Quando perceber, já era, né? Eu acho que tem isso, de ver.
Ballenas, delfines, corales, está lleno. Lo ves más las ballenas que los pandas, ¿eh?
Pero no, tú lo ves como
hay más gente que conoce la ballena que
lo ves como algo anecdótico. Tú no lo ves como algo. Tú ves un árbol y el árbol dices, hostia, esto me está ayudando. Pero tú una microalga, fitoplancton, eso no te ayuda a la mierda y te está dando el 50% del oxígeno del puto planeta. Pero tú no lo ves y no lo ves y dices, pues esto no, ¿qué es esto? Tengo un meme genial de las algas que ponen, pues eso, todas las microalgas diciendo, cuando los árboles se llevan todo el mérito del oxígeno, siendo las microalgas las que tienen realmente el papel principal. Pero es que es dramático, en mi opinión. Entonces, ahí hay un tema de distancia. No nos vemos, no nos estamos identificando con el océano.
É um ótimo debate, né? E eu acho que eu sou o público, meio que eu estou entrando agora na carreira acadêmica, né? E eu acho que eu sou o público que vocês estão querendo chegar, né? E, na minha concepção, eu faço parte de um programa de educação ambiental marinha, né? E o que eu mais vejo das crianças quando chegam perguntando, quando eu mostro alguns animais, várias crianças, isso aqui eu como, esse aqui é sempre eu como ou eu não sei o que é, o animal estranho. Eu acho que essa relação do porquê que essas pessoas não enxergam o mar é justamente pelo fato da cultura que a gente coloca sobre o oceano, de que ele é um produto que a gente tem que consumir. E tanto o peixe quanto os animais bentônicos. Por exemplo, antes de eu entrar na faculdade, animal bentônico, o que é isso? Para mim era tipo assim, era camarão e... Não, camarão não. Não que a gente come caranguejo. Para mim era caranguejo que servia para alimentação. Eu não sabia o que ele fazia, qual era o papel que ele fazia, ou o que ele tinha com o ecossistema. E eu acho que as pessoas, quando estão no mundo aí, justamente o que o querido falou, é que, tipo assim, essa relação é muito mais, pelo menos aqui no Brasil, também, né, em outros países devem ter, que a gente tem que ter a nossa sobrevivência e depois pensar nessas questões ambientais. E a gente tem que ter essa correlação das pesquisas com o público. Por isso que eu acho que o papel do pesquisador não é só fazer um projeto e compartilhar com pesquisadores, porque, na minha concepção, o que importa uma pesquisa se só quem vê é outro pesquisador, já que quem, teoricamente, deveria saber que faz a mudança é a população. Como é que isso chega? Então, eu acho que pesquisas voltadas para a área ambiental, no quesito de conversar com pescadores, ensinar, fazer workshops ou palestras ou uma conversa mesmo, que eles também têm conhecimento que possam nos entregar, e a gente também tem que fazer por eles para que consiga ter realmente uma boa preservação ambiental, porque senão como é que funciona, como é que o mundo vai mudar se a massa consumidora dos produtos pensam de outra forma?
Eu acho que entra também muita a questão da fofofauna também, principal tanto terrestre quanto oceano, porque tem essa afetividade. Eu acho que isso é muito importante, porque no terrestre a gente tem a afetividade com gatinho, cachorrinho, ararinha azul. Agora, quando você vai para o oceano, pelo menos dentro do meio que eu vejo, sempre ou aquele animal de consumo ou aquele animal feio. Não, a tartaruga até que o pessoal acha bonitinho, né? Mas a maioria, assim, eu vejo que o pessoal tem uma certa aversão, assim, não tem esse carinho que eu vejo. Vamos defender os animais terrestres porque são bonitinhos. O oceano eu já não vejo tanto isso quanto nos animais terrestres. E aí eu acho que isso também impacta um pouco, porque a gente costuma querer defender aquilo que a gente sente uma certa afetividade. É tanto que uma das cadeiras que eu fiz na faculdade, a gente começou a pensar num projeto de defesa de golfinhos. E aí, para a cadeira. E aí eu vi que todo mundo começou a se apegar muito à história do golfinho, porque deram um nome para o golfinho, fizeram toda uma história. Então, quando a gente traz essa afetividade, quando ele começa a fazer parte do nosso dia a dia, aquele animal, o oceano em si, ele começa a ser importante, a gente quer preservar aquilo que a gente gosta. Então, eu acho que ainda também é muito dessa estranheza, essa distância que as pessoas elas têm com o oceano, que elas não têm com o ambiente terrestre em si. Porque vai muito com o que a Carol falou, o ambiente terrestre a gente está aqui, a gente está em cima, a gente vê. O mar já tem aí essa distância a mais, aí eu acho que essa questão também impacta um pouco.
Mañana a Juanita falará por la tarde para una palestra de eso y después faremos una experiencia precisamente de comunicação, de engagement, de opinião, etc, etc. Eu penso que será muito interessante ver esse tipo de... Mas sim, é assim mesmo, é complicado. Às vezes, eu vejo que quando você fala como experto e você tem uma audiência, os seus amigos, a sua família, gente que está próxima, estudantes, alunos pequeños. Eu, por exemplo, eu he feito aulas a todo tipo de pessoas, desde pessoas que estão já retiradas da vida laboral a niños de 3, 4 anos. Você busca um tipo de linguagem para cada um. Perfeito. Eles estão interessados nos oceanos e estão fascinados por os oceanos. Mas para ellos los océanos es una cosa que está ahí, que está bien. Y está bien ahí, pero que no tan bien. Pues más, puede ser que sí que sea más importante, no sé, los árboles que dan fruta o, no sé, las vacas que dan leite y cosas por el estilo. Ellos tienen una visión que es o que você decía, o sea, sí, pero ¿cuál utilidad de todo eso, no? Y hacer comprender a gente o que está pasando e a importância de preservar os oceanos com os meios terrestres também é complexo. É complexo. Bom, ¿tienen más preguntas, más intervenções? Sí. Vale.
Boa tarde. Eu queria saber se, no contexto das MAFs, se já tem alguma discussão relacionada a estratégias de mercado que influenciem ou que tenham como foco a preservação, que é uma discussão que a gente tem atualmente com os manguezais, bastante grande, né? A questão dos créditos de carbono e de crédito de emissão de redução também. Os manguezais têm sido bastante visados, né? Aqui no Brasil a gente tem estados que estão fazendo inventário de créditos de carbono disponível nesse ambiente. Então, eu queria saber se nesse contexto das MAFs isso já é algo discutido, se já tem interesse por setores privados e públicos da utilização desse recurso para esse tipo de finalidade, né? Estratégias de conservação a partir de financiarização dos serviços do ecossistema.
Esa pregunta es directamente para mí. La última slide que no puse, porque era muy larga, de la minha presentación, era exactamente eso. Mas eran créditos de biodiversidad, que es una cosa que surgirá. Seguramente será presentada en Belém, en la COP30 de Belém, que hay mucho interés en los créditos de biodiversidad. Se está pasando de crédito de carbono a crédito de carbono biodiversidad. Y nos publicamos un paper ese año de créditos en Ecology and People, Ecosystems and People, Ecosystems and People, una revista de Wiley, una buena revista, falando de eso de la MAF, de la Marine Animal Forest. Ninguém ha feito nada de crédito de biodiversidade no oceano. Ninguém, você pode buscar. Porém, Biodiversity Credit, Ocean, o Biodiversity Credit, Marine Forest, Biodiversity Credit, Ventos, Biodiversity Credit, Seas, nada. Solo un paper, o nosso paper. Que aí está a pergunta, quem paga? Porque eu quero que se faça um plano muito sério de conservação e restauração real, com muita grana, pero quem paga? Porque na Europa, por exemplo, han dito, no, espera. As arcas públicas não têm esse dinheiro, não temos dinheiro para fazer isso. Por isso, nos projetos europeus se estão fomentando os programas mixtos público-privados reais. Uma coisa real, público-privada, onde eu investe, o persona que pone dinero. O que pasa es que eso es peligroso. Yo tengo todo un raciocinio, tengo toda una presentación de eso, onde falo de que o importante é facer un traballo moito serio, transparente, onde você calcula a biodiversidade e esa biodiversidade pode ser cuantificada e esa biodiversidade você pode ver se hai unha progresión positiva ou non. Mais, gente que hai aquí, como Tito, como Lorenzo, y personas, obviamente, como los 3 palestrantes que hemos tenido, que Américo, Carol y, por supuesto, Martina, están trabajando directamente en biodiversidad. Entonces, o que nosotros hemos enviado ahora, o segundo paper da revista que hemos enviado ahora a One Earth, que es una revista también bastante interesante, en One Earth nos hemos enviado o que nos pensamos que podría ser o comienzo de los Biodiversity Index, que son índices que pueden ser fácilmente, digamos que clasificables, pueden ser fácilmente cuantificables por un tercero, por un auditor para fazer realmente, isso está aumentando a biodiversidade. Mas, qual é o problema? É que a biodiversidade é tão complexa, que esos índices que nos estamos presentando son solo o inicio pequeñito, pequeñinho. Porque você aí não está incluyendo cosas como a biodiversidade de que eu estou trabalhando, funcional e trófica, por exemplo, porque é demasiado difícil ainda de ter um consenso. De isso é muito complicado, complexo. Mas começamos com isso. Por quê? Que me interessa a mim. A mí me interesa, y me interesa de verdad, que o capital privado pague. Él tem que pagar. Él tem que fazer isso. Mas se você dice, você tem que pagar, então temos um problema, porque o capital privado faz, não, espera, não. Eu não quero fazer isso. Se você faz uma outra estratégia com o capital privado e diz, não, espera, vamos ver. Você ha entendido o conceito de biodiversidade? O 99% te dicen sí, e o 99% não ha entendido nada. Não sabe o que é a biodiversidade. Quando você explica, começam a estar preocupados. E o que eu posso assegurar a você é que muito deste capital privado começa a compreender que o problema é muito mais sério do que parece. As pessoas que estão nas empresas não são de evil. Não, são pessoas que têm outra mentalidade completamente diferente. Se você tem a capacidade de fazer compreender a elas que realmente a sua própria supervivência está on the stake, ou seja, está en peligro, então es cuando esas personas comienzan a decir, ok, un momento, vamos a hacer alguna cosa. Hay, que yo recomiendo muy ferventemente, hay un divulgador científico del CSIC que se llama Antonio Turiel. Antonio Turiel es un físico oceanógrafo que trabaja básicamente en la circulación oceánica. Él trabaja en tres grandes divulgaciones. Uno, a falta de recursos energéticos, petróleo, gas y carbono. Y él demuestra, básicamente, no solo él, que estamos yendo hacia un declive real de lo que serían las energías no renovables. O segundo foco de interés de Antonio Turiel es a mudanza climática, demostrando que a mudanza climática está afectando y tal. Y el tercero es las corrientes oceánicas, obviamente. O que Antonio Turiel ha visto es que él hace mucha divulgación, tal, vale, tiene una manera de explicar también bastante pesimista, vamos a ser realistas. Mas lo que él dice es que durante los últimos 5 años, los empresarios llaman a él para sentarse y decir, por favor, explícame qué está pasando. Y él explica con simplicidad. Y mira, pasa esto, pasa esto, pasa esto. Y los empresarios son los primeros que comienzan a decir, aquí tenemos que nos adaptar. Si no nos adaptamos, colapsaremos. Por eso, y repito, y es una cosa que va hacia adelante. Entonces, a mi opinión, es que evidentemente hay mucho tipo de empresario. Hay empresarios que no importa nada, o extractivista, o gente que está en otra esfera. Yo pienso que hay gente como Elon Musk, o Mark Zuckerberg, que viven en otro mundo, no viven en la Tierra, viven en otra historia, tienen una fantasía. Ellos tienen esa fantasía, ¿sabes? Mas hay mucho empresario, mucho empresario, pequeño y mediano empresario, y de empresas que comienzan a comprender que la cosa va también con ellos. O sea, es un problema también de sus familias, de sus amigos, de sus amistades, de su mundo. Entonces comienzan a decir, un momento, vamos a ver cómo podemos revertir esto. Por eso, sí, mais ainda está verde. É como un mangue, mas muito verde, que es imposible de comer ainda, a pasagem do crédito de biodiversidade real a una parte aplicativa a través de esos índices. Nos estamos comenzando a explorar, como cientistas, estamos comenzando a explorar. ¿Es posible que al final no cabe nada? O sea, que al final aquello no. O es posible que a gente comience a animarse y comiencen a ver otros grupos, otros cientistas de las diferentes partes del mundo que comiencen a decir, un momento, vamos a hablar de eso. Yo creo que podemos mejorar este índice de esta forma. Vamos a intentar complementar este índice con este tipo de información. Vamos a intentar hacer más transparente este tipo de índices con este tipo de procedimientos. Puede ser o no, no sé. Vamos a ver. Mais perguntas? Mais intervenções? Então, até amanhã. Vamos vernos mañana, ¿ok?
Oi?